Oh my freaking goodness, he did it. The madman did it. I can't even believe it. I gotta listen to myself. The madman did it. Oh yeah, I did. I just heard myself say it. What is up, beautiful people? Look, look. I know what you're thinking. I can't believe it. He's early. This never happens. Well, it just did. Just freaking did. What's up, couple fish? Yo, this is finally a stream. We talk about something educational. We never do that around here. You know, it's always bacteria cultures, sponge filters, breeding fish. This is ridiculous. We can't learn from any of this. Holy fish capes, what's up? Stream, we talk about something educational. Yeah. Yo, did anyone read the description? I put a lot of effort into this, okay? I wrote a poem for this stream. This is gonna be the Loach research paper stream, all right? I actually did my homework, okay? I lined up all the articles. I haven't read most of them myself. I just a short scan. But, you know, I was so in the spirit when I decided to do this stream. I wrote a little poem, okay? And I had a beat in my head when I was doing it. It said, put me in, coach. Never fly coach when I'm picking up the loach. Put me in the game, swear to God I rock the boat. Best sucker fishes, you know, loaches get the vote. Yeah, I'm instigating, yeah, you know I'm gonna poke. Bears hate to see when we go live and push the yoke. I'm pushing the line, it's fine, we got the load. Pseudogastromyzon, we with the UFOs. Oh my God, bro. Oh my God, bro. We with the UFOs. So, more like the next fish rap. No, it's a poem. It's a soliloquy. It's a, what's it called? It's a haiku. It's all those things. You know what I mean? What is up, sweet Koyo? TK Tropicals. No Bloody Mary Loaches? What? Why not? Mick of course, the platypus. A couple people were checking in the chat early. Jesse, how's it going? Yo, 1 loach, 2 loach, red loach, blue loach. Heard tropical fish soon gonna get a new loach. See what I mean? Yo, speaking of Tropical Fish Co., just real quick as we about to dive into this. But speaking of Tropical Fish Co., dog, they just blessed my club, the Aquarium Society of Central Maryland. Yo, I could not believe this. They donated. I gotta make sure I get this right because I honestly was in shock. I still am a little bit, but shout out to Tropical Fish Co., where my club is that I'm a part of is doing a swap this Sunday, and they donated 3 $100 gift cards to our raffle. It's just crazy. I've never even seen $300 before. But yeah, if you're in the area, we got some crazy raffle items loaded up. And this is just it's top. I was already pretty excited, but this is just like ridiculous. Okay, so come through this Sunday. You can find the details, man. Aquarium Society of Central Maryland. Sage Case of Wonder said the 6 tiny baby pea puffer came out hiding today. I thought I lost them. I'm glad that they're still around. Good. Okay, so let's just go ahead and hop into things, because I thought usually when I start early, people aren't here, like they're like not expecting it. But you guys are actually on top of things today, so well, let's hop into stuff. Let's hop into some stuff. We're actually on top of things today, so well, let's hop into. I'm checking my audio here. Okay, so I decided I wanted to learn more about loaches recently, so I went on a bit of a spree of loach research, and I learned a lot of different things. Just reading the titles, I learned a lot of different things I was not aware of when it comes to loaches, such as the fact that some of them are happily consumed in large quantities. Okay, this is not something that I was aware of. They're actually aquacultured for food in places, so that was new. I never thought of eating a loach snack pack, but here we are. This is a thing. Let's hop into some stuff. A couple of fish says, yeah, my loach book has recipes for gastros. Wow. Okay. So you have recipes for loaches.
All right, I'm not sending my loaches to your house. That's for sure. That's for sure. Okay. But besides them being consumed for food, I found cave loaches. I found loaches that of unusual size. No, I'm just kidding. Um, I found they were testing the effects of taurine on the loach immune system. Oh my gosh, man. I found that birds, apparently some birds be eating loaches out there. I never even thought about what was eating the loaches, much less the loaches themselves being eaten. So this is something that kind of did take me down. It took me down quite the path. So let's get into this. First up, I want to get into this new cave loach species because I think new species is kind of exciting. There's a couple new loach species, but the one that stood out to me first was the cave one because I kind of had some cool photos of it. So let me just boop, 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 find my freaking window tab. Here we go. Getting a fast coffee. BRB. Okay, Mick, take your time. Take your time. All right. So over here, we got the chromosomal level genome assembly of the obligate cave loach. Okay, that's one of the exciting ones, right? That's not the one I'm looking for, believe it or not. Here we go. Here we go. A new typical cave fish of the genus Triplophysa, Telosti, Crypniformes, Nemochilidae from the Jinsha River, Yunnan, China. Now, I recognize this name, the Jinsha River, because I feel like there's a loach they call the Jinsha loach, I feel like. Yeah. And this is one that I believe that this is one of the fancy ones that that Jody has or has acquired at some point that look kind of nuts. So I thought of that name immediately when I saw the Jinsha River. I immediately thought about that. Yo, what's up, Jody? What's up? A couple fish said Cryptotora, baby. Okay, Cryptotora. Water climbing cave fish. Okay, okay. Interesting. All right, well, new loach just dropped. A cave fish. Triplophysa Vichingensinensis. Oh my God. They really think that I'm going to be able to read this out loud? Why didn't they consider me when naming this fish? I'm pasting the name in the chat. You guys can try it for yourself. A new cave-dwelling loach was collected from an underground river outlet in the Xingangshi village, and it's in a reserve for endemic fish, part of the Hejiang-Jinsha River system. The new species is distinguished from congeners by significant genetic divergence and the following combination of characteristics. I'm so glad that they're including something besides the genetics that I can understand. Jody said Gene Shy Shinensis. I was hoping to get Gene Shy abrivida on the last import, but that didn't get filled. Oh, unfortunate. Okay, so it's distinguished by its vestigial eyes. Okay, so I guess that's, let me, vestigial, yes. Something that was once much larger. Okay, these eyes are no longer needed. Absence of skin pigmentation, pelvic fin tip reaching the anus, complete lateral line, developed posterior chamber of the swim bladder, and dorsal fin rays 3 to 7. So, apparently, Triplophysa is the most species-rich genus in the family Nemochilidae, comprising of 166 valid species with 78% distributed in China. They are primarily distributed across the Qinghai Tibetan Plateau and adjacent regions, but are also found in southwestern and northern China. The morphological characteristics distinguishing them from other genus in the Nemochilidae include anterior and posterior nostrils, closely set, a posterior wall of the bony swim bladder capsule, body cylindrical anteriorly. They're just making this up at this point. Progressively compressed posteriorly and sexual dimorphism, tubercles and thickened pads on the snout, cheeks, and dorsal surfaces of the pectoral fins. Howe et al. In 2010 categorized these into 12 distinct types, notably of which one demonstrates sexual dimorphism in the head and snout shape. Males possess a squarish snout and dorsal view, while females exhibit a round snout. We love a sexually dimorphic loach. That's kind of like what I like to hear about the most. Okay. So this species is classified into hypogean, parentheses, cave dwelling, and epigean, which is surface dwelling groups based on ecological habitats. Let's see how they caught these guys. Let's see. Excuse me. Okay, specimens were collected using dip nets at a subterranean river outlet. Samples were either fixed in 75% ethanol or transported live to the laboratory. Oh, good. They took some of them alive before killing them. After the live photos were taken, live samples were fixed in 10% formalin. What's up, SBC? Where's the fun in dimorphic? I think that there's plenty of fun to be found. You can find fun anywhere you look. You know. Before fixation, the right pelvic fin rays were clipped from all samples for molecular analysis and preserved in anhydrous ethanol. Okay. Measured with digital calipers. Well, that's what we like to see. Okay, they're doing it the right way. And here we got a list of some species and the results. So this is something that was kind of cool. So we got the hypogean and the epigean groups of this species, the Pseduogastromyzon. And you can see how many of them are in the hypogean group. Seems like a large majority of them seem to fall under this. What is trogloscenes? What is that? Trogloscenes or subtroglophiles are also called cave guests. They periodically live underground but cannot live exclusively in such habitats. Okay, so temporary cave dwellers. So if you're someone that spends a lot of time, in your room, and your room happens to be in a basement, I'm not calling out anyone specifically, but you might follow under this trogloscene group. Okay, so I'm gonna have to remember that for myself. Let's look up the name of one of these epigean groups. Triplophysa stolixi. Triplophysa Stolickzi. I just read that whole thing out loud and forgot it immediately. Hopefully one of you remembered. Okay. Interesting. This Tibetan stone loach. That looks like. I mean, color-wise, it's not too much. I mean, this picture looks a little bit cooler, but look at it. It's got like a tummy on it, man. Look at these things. Look at that beefy midsection on some of these guys. Yeah, this is the right loach. Why did they just toss it out on the rock for this photo, or does it go out the water? Because it kind of looks like they just tossed the damn loach on the rock for this picture. Oh my God. Okay, well, rest in peace, loach. What up, Big Steve? How's it going? How is it going? Okay, let's take a look at where they found this thing. So I think the coolest part of this paper, honestly, isn't even about the loach necessarily. It's about how much information on all these other loaches and where they're at is kind of fascinating. Oh, let me put the link to this in the chat. Because you can just see how many different species are all over, and they got the clade 1 and the clade 2 up in here categorized. What's up, Johnny Best? How's it going? We'll take a look at these cave loaches. The vestigial eyes, quite disturbing. What's up, JP Jeffries? Artist working in the building. Yeah, I can't tell if I love this or if I'm intimidated by it. I appreciate the X-ray. I do. I do appreciate the X-ray. So here you can see what you're talking about with the snout being asexually dimorphic. So the female on the left, see it's a bit more pointy. We got the square snout on the right. It's the male. This would be really, man, I would, if more loaches were like this level of ease and telling the difference. I mean, look, even like how the head's like more concave on this side on the male is kind of useful, accessible. That's the word I'm looking for. Like someone like myself can actually tell the difference, and I appreciate that. Okay, I'm just moving my thing around. So here is where they did the dip nets at on the side. So it must be some underground cave entrance right here. This is a beautiful looking place, man. Though, like this. This looks like a scene from a movie. Like they're stranded on an alien planet that's Earth-like. Jody said most females will be fuller in the abdomen. Quotation most. Yeah, I need a dimorphic snout. Okay, leave the abdomen shit. That can be for people like you, Jody, who have some kind of expertise. I need a snout. Yo, my chick's gotta have that oval snout, man. This is the line I'm drawing. Okay, this is a live photo of the Triplophysa bishingenensis. The red circle indicates the subterranean river outlet. Okay, so this is the subterranean river outlet where they did the dip nets at. And then you can see here in the lighting with some nice rocks behind it, which check out these rocks, man. These rocks look dope. Yo, I need some gravel. I need more gravel like this. My next tropical, well, my first tropical fish order will contain loaches. I'm gonna have to I'm gonna have to make a specific tank to put them in, or just one I have now. I gotta have some freaking gravel like this. We're gonna go through some of these river things in these papers and get some inspo. But in this lighting, this fish looks pretty cool. In this lighting, where they've killed it and then put it in ethanol and stuff. I mean, we talk about the fish not coloring up in the bag. The colors in the lab and in the preserved specimen jar are not hitting either. So thank you for the live photo. Sutton said, finally a YouTube on with knowledge. What's up? Yo, you heard? This is our annual stream with vestigial educational content. Camjan said, "What the heck? I thought it didn't start till later." Hi guys. You guys didn't. You don't. You guys got to read my descriptions. I put in the description that it might start earlier. Okay, but you can thank Jody for making sure that happens because she did ask in the Discord if I could start earlier, and that's why you should be in the Discord because sometimes the pleas do make a difference. Okay. Um. So the whole body lacks pigmentation and scaleless. Live specimen, semi-transparent, flesh-colored, internal organs, vaguely visible ventrally. I don't think we got a perfect ventral view here, but I mean, yeah, you could kind of see some organs up in there. Yo, Mick with the long hello. Joey says it's because I'm lame and have an early bedtime. Yeah, 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 you got to stay up, man. Yo, I'm about to get another cup of coffee right now. Yeah, eyes vestigial, reduced to black pigment spots. 2 chambers of slim bladder. Anterior chambers dumbbell shaped and encased in a bony capsule. Okay, okay, interesting. After fixation, body turns white, abdomen pale yellow, eye spot color intensity varies with some nearly individual. And then here we have the sexual dimorphism. All specimens lack thickened pads and furcella on the head and pectoral fins. However, male and female show significant differences in head shape. Male heads narrow sharply at the eye position, then taper gradually, resembling a bell shape with a blunt snout. Female heads taper gradually, resembling a triangle with a pointed snout. Yeah, all my females need a pointed snout, man. I'm looking at you. Looking at you. Yeah, even you over there, fish. We need more fish beauty standards, fish plastic surgery. Forget injecting dye. When are we going to get the snout plastic surgery for a fish? Uh, Cam Jam said, "Who's your daddy, Mickham?" What the heck? Um, okay, here we go. Currently documented cave-dwelling species of Trypophylla are predominantly distributed in the karst landscapes of eastern Yunnan, northern Guangxi, southwestern Zhejiang, and the Hunan border region. Hydraulically, with the exception of these other species within the Red River system, all documented species inhabit tributaries of the Yangtze River. The Yangtze River harbors 7 species compared to the 33 species in the Pearl River system. Its biogeographical pattern marks spatial heterogeneity. The Yangtze system demonstrates lower species richness with scattered distributions, whereas southern populations exhibit higher species diversity and aggregation density. This pattern likely reflects differential development of karst geomorphology between regions or uneven historical dispersal processes. Okay, okay. Mm. The Trogloxine group, sister to the Hypogean group. So the Trogloxine group, those are the ones that can only live in caves part-time. And then the Hypogean group are the ones that do go above water. Bro, really in his lost Chinese area with pronunciation like that? Listen, set your expectations low, okay? I can barely read English, but I'm trying. I'm trying, okay? I'm trying. I mean, I'm skipping a lot of words too, but sometimes I do try. Mick said, Nick is my daddy. Pause. Nick, you're like, you're a little bit too old for me to be your father, and unless with time travel's coming into the play. Okay, so the group that sometimes lives in caves is sister to this Hypogean group and is currently only found in the Upper Yangtze regions as a lineage embedded within tribal Physa. Okay. Claya retains generic validity due to the absent secondary sexual traits and processes. Dentiformis on the upper jaw. Current evidence thus suggests the polyphile of this group with more than 160 documented species. Oh my God, taxonomic revision. This is getting too deep for us, I'll be honest. Among the 43 cave-dwelling species, 16 species inhabit cave entrances, subterranean river outlets, or surface water bodies connected to subterranean systems. 77% retain normal or partially degenerated eyes. Okay, notably this one, despite exhibiting extreme ocular degeneration and pigment loss, retains the ability to survive briefly outside the cave near its outlet, suggesting that some cave-dwelling Triplophisa species retain limited adaptability to surface environments. These species may periodically leave caves independently or be displaced by flooding events, resulting in short-distance dispersal to other suitable subterranean habitats. Interspecific genetic differences among cave-dwelling Triplophisa are relatively low. Oh man. So this is the first recorded cave loach in the Yangtze River system basin within Yunnan, and simultaneously the first documented typical cavefish in the Jinsha River system. Its discovery validates the presence of environmental conditions conducive to cavefish evolution in this basin. Field investigations conducted by authors reveal extensive karst development within this river system, exhibiting particularly dense subterranean cave systems and underground river networks, highlighting the potential cave biodiversity in these areas and warranting further investigation and research. So there you go. New typical cave loach. We're starting off strong. I mean, this guy does look pretty cool, but probably not a good aquarium fish. Sutton said Mickham's father was live. Oh, okay. If Nick says jump, I ask how high. Okay, let's see. Can we get a 3-foot jump from you, Nick? 3 feet. Is that too much to ask? Is that too much to ask? I'm asking for a friend. Just asking for a friend. All right. Well, pivoting a little bit. Remember how I said about loach aquaculture? Tell me if this looks familiar to you. And if you're sensitive to anything, I apologize in advance. Hold up. This page refuses to load, apparently. Hold up. Hey, nerdy knowledge given for free. Count me in. No, there's a cost. You have to listen to me mispronounce everything. So this is fried dried loach, 500 grams, ready to eat, spicy snack, side dish for drinks, specialty, and leisure. Yeah, loaches as a snack is like a big thing, apparently. Yeah, so you could just buy some fried loaches in a snack pack. I know it's hard to fully picture. It gets even worse. It gets even worse, man. You recognize these guys? Yo, I bet they're not charging $100 a meal for this snack. You recognize these? Okay, so you know there's some aquaculture opportunities for some of these fish where we look at them as pets, and some of them are relatively expensive over here. They'd just be snacking on them bitches. Okay, snacking. Axolotl jerky sounds pretty disgusting. I'm not gonna lie. That I'm not on board with that as much. I'm really not. I think it's the jerky part. I think is probably more I think that's probably more the issue for me, the jerky part. That's kind of challenging for me. I don't know about you, but yeah. So I had to kind of dive down this a bit. Zebra Aquatic says YouTube isn't notifying me of streams. Yeah, I need to add a notifier in the Discord more because YouTube doesn't do its job for sure. Food, why now? I'm hungry. Well, you're hungry now that we're looking at the fried loach? Really? All right. Everyone write that down about Moneyless Maven. Don't take your fish around them. If you have too many loaches and you're like, I just gotta get rid of these loaches, I got space for them somewhere. You know exactly what's about to happen to them. They're about to eat the loaches. Holy. Okay, so that leads me into. Is this the one that I'm looking for? Let me double check. Let me double check. I had some aquaculture. This is kind of a cool fish. That's not the one I'm looking for though. I buck tradition. I'm a woman who eats. What? All the women I know eat. Pause. I don't know about you. Eating tends to be kind of necessary, but I respect that you buck tradition. That is good. Well, besides the fact that it's kind of interesting that they aquaculture these fish for food, this, of course, led to the fact that there's some interesting research papers revolving around loaches and preventing them from dying uh, more due to the fact that their death might impact profit margins when it comes to food farming. So I found this paper here, and I'll put this in the chat. The effects of taurine on immunity and ammonia metabolism in large-scale loach under ammonia stress. Okay. If they eat Corys as the last straw, I hope they don't. We're really picking and choosing here, huh? So ammonia is a common environmental pollutant that's extremely toxic to aquatic animals. Therefore, there's urgent need to increase ammonia tolerance in aquaculture animals to achieve high-quality development of the industry. 3 treatments were designed to examine the effects of taurine on the large-scale loach. Now, taurine, we'll just do a quick, just a refresher. What's something interesting about taurine? Mayo Clinic. Taurine and energy drinks. That's where I know it from. So what does the Mayo Clinic say about taurine as a food item? Taurine in certain amounts is safe to drink. It occurs naturally in foods with protein, such as meat or fish. The human body uses taurine for actions and cells. One example is that taurine is used for energy production. It also helps the body process bile acid and balance fluids, salts, and minerals, among other actions. All right. So that doesn't sound like too bad of a thing. Taurine. Well, there was a control group exposed to water and injected with physiological saline, an ammonia treatment group exposed to 3-MOL/LNH4CI solution and injected with physiological saline, and a taurine treatment group injected with taurine. Immune and ammonia metabolism related markers were measured at 12 hours, 24 hours, 48 hours, and 96 hours after treatment. And let's see what went down. We got a graphical abstract. Hold up. Let's look at this. So ammonia stress. This is a fantastic flow chart. Okay, here we go. Ammonia stress. Okay, so the ammonia stress is entering the loach. Then over here, the alleviating effect of taurine. Regulate glucose metabolism and osmotic pressure balance, release stress, restore normal blood sugar and osmotic pressure, mitigated immune stimulation, enhanced ammonia tolerance. All right, so these are meeting together. The loach under ammonia stress meeting the taurine injection. And this is going over to the gills and the gut. And we have the liver and kidneys here watching on with horror as this chemical experiment proceeds. But taurine enhances the ammonia tolerance of large-scale loach by regulating immune response and promoting glutamine synthesis. Okay, this is high promises right now. The graphic already got us off to a great start in this one. We should feed our fish Monster energy drinks. It's kind of what I'm getting from this, right? Modern aquaculture facilities and techniques are commonly used to rapidly remove ammonia, nitrogen, and organic matter from water bodies such as aquaponics. Another strategy is to increase the environmental ammonia tolerance of aquaculture species. Like, yeah, one strategy is to try and remove the toxins. The other strategy is just to improve the fish's ability to survive in a toxic environment. Now we're talking. This is primarily achieved by exogenous addition of substances that can boost ammonia detoxification, such as taurine, arginine, and peppermint oil. What the hell? Okay. Write that down for later. Peppermint oil. Apparently this is, maybe we all should really actually be buying all the what are the fish meds, or like some kind of tree oil type stuff, Primafix and all that kind of stuff. I forget. The addition of additives is simple and practical, and therefore widely used in production. Taurine is sulfur-containing, conditionally essential amino acid in fish and crustaceans, as its dietary requirement varies significantly among species. It plays important roles in the growth, feed utilization, antioxidant capacity, stress resistance, and immunomodulation of aquatic animals. Okay, so this is all them justifying the study. Let's scroll down a bit. The large-scale loach, paperszzz dabrianus, is an economically important aquaculture species in East Asia. Let's see what loach we're talking about here. Okay. Let's see what loach we're talking about here. Paperszzz. Yeah, this looked like a big old loach. The large scale loach is really what it's called. Really. Damn! Oh, yep, that's a large scale loach right there. I see why they're frying them up like French fries. I mean, they're the perfect shape. They're actually kind of the perfect shape. Let's see what the USGS says about this. Man, the government website, it's loading like molasses right now. Come on. Okay. We'll have to spin back to that. Let me just look up, because I'm seeing the food, the fish as a food, and I'm trying to see the fish as a living creature, prey food. Yeah, okay. The large scale Loach. A seriously fish. Got a page on them. Subadult specimen from the aquarium trade. Okay, so they make their way into the aquarium trade, I guess. That is crazy. Okay, back to reading. So large scale roach is loach. I said the large scale roach is an economically important aquaculture species in East Asia. As a typical air breathing fish, although the large scale roach has an extremely high tolerance to environmental ammonia, ammonia can still stress and suppress its immune system and antioxidant capacity. Due to its unique physiological characteristics, intensive farming and rice field culture have become common in large-scale loach production. High density and small water body environments cause large-scale loaches to encounter ammonia stress during aquaculture. All right, we got the ethics statement. They purchased the fish from an aquatic product market in Hefe and temporarily acclimated them for 7 days. Okay. Oh, I'm on the wrong page. That's why I'm not seeing the whole thing. Give me 1 second. I need the full text. I need the full text. I clicked on my own wrong link. Here we go. Here we go. I'm posting this new link in the chat. Okay. Cookie settings, only necessary. Open PDF in browser. We're back. Where was I? It's so hard to read. You ever notice that? The difficulty? All right, we got some more figures here. Okay, after exposure to ammonia for 12 hours, all right, gill AQP3 expression was unregulated to 2.65 times that of the control group. Okay, the taurine treatment inhibited the ammonia-induced upregulation of gill AQP3, and AQP3 expression was significantly lower than in the ammonia treatment group. The expression level in the kidney increased initially before decreasing under ammonia stress, reaching a peak at 24 hours, but the expression level was significantly higher than that of the control group during the entire course experiment. Okay. Compared to the ammonia treatment group, taurine inhibited the upregulation of kidney AQP3 and showed a similar variation trend of initially increasing before decreasing. At 96 hours, kidney AQP3 expression was lower than in the other two groups, with increasing duration of ammonia exposure. All right. Ammonia nitrogen is a common environmental toxin in water bodies, and a large volume of research has shown that ammonia accumulation can suppress the immune system of aquatic animals. Ammonia exposure can induce the production of reactive oxygen species in aquatic species. With the accumulation of MDA, the body will usually increase the expression of antioxidant enzyme-related genes to scavenge excess ROS, the reactive oxygen species. Taurine is a scavenger of free radicals such as hydrogen peroxide, superoxide, and pyronitrite. And exogenous taurine can similarly alleviate ammonia accumulation-induced reduction activity in fishes. Okay. In this study, intraperitoneal injection of taurine increased SOD activity in large-scale loaches exposed to ammonia. The complement system included many plasma proteins involved in the elimination of cell debris, preventing pathogen invasion and alleviating inflammatory responses. Man, they make it sound so fancy. Like, this sounds like the best thing ever. IgM activates the complement system to exert non-specific immune effects such as bacteriolysis, bacteria killing, phagocytosis, and agglutination. In this study, exposure to high concentrations of environmental ammonia did not significantly affect the liver IgM or C3 contents in large-scale loaches, but significantly affected the kidney IgM and C3 contents. So the liver is cool, the kidney, not so much. This may be because the kidney is an important immune organ in fish. I see I'm dropping. I gotta move myself here. I just started randomly dropping some frames. Its important immune system in fish and its response to ammonia exposure are more direct. Previous studies have shown that accumulation of ammonia decreased IgM and C3 contents in turbots, yellow catfish, and other bony fishes. Okay. Okay. Lysozyme is the first line defense of the innate immune system in fishes and is primarily located in the body surface mucosa and kidneys. Lysozyme disrupts the peptoglycan layer of bacterial cell walls, leading to the lysis of pathogens. As such, lysozyme plays an important role in the defense against bacterial and viral infections in fish. In this study, ammonia exposure significantly induced LYZ expression in the liver and kidneys. Ammonia and high temperature stress decreased LYZ gene expression in Nile tilapia. Here, taurine injection restored LYZ gene expression in the somatic tissues of large-scale loaches to levels similar to the control group, demonstrating that taurine could alleviate the immunostimulatory effects of ammonia. Okay, this is all about the immune system. Similarly, ammonia exposure induced the expression of heat shock proteins in obscure pufferfish to protect the body from oxidative stress caused by ammonia toxicity. The heat shock proteins can activate dendritic cells and macrophages, promoting the maturation of major histocompatibility complex molecules. Therefore, overexpression of heat shock proteins can damage fish. Interesting. Now, is there cold shock proteins? That's not my immediate question. In this study, exogenous taurine induced liver expression in large-scale loaches under ammonia stress, but inhibited kidney HSP70 expression. This suggests that the increase in liver HSP70 did not reach the threshold for damaging hepatocytes and thus represents a strategy for boosting immunity. Okay. In contrast, downregulation of kidney HSP70 expression is a form of negative feedback. In addition, the reason for the downregulation of HSP70 expression in large-scale loaches in the later stages of ammonia exposure may be that the downregulation protects tissues from injury due to excessive heat shock proteins. Wow. This is a lot to take in right now. It's a lot to take in. Heat shock proteins. Okay, I'm gonna write heat shock proteins down because I need to look at that later. Okay. Heat shock proteins in fish. All right, guys. Because understanding the fish immune system better is, that's on the to-do list, and I feel like this is some interesting stuff here. Okay, let's see how it affected the ammonia metabolism in large-scale loaches under ammonia stress. Big Steve said they're proteins from heat shock. Okay, fucking genius in the chat. Thanks, Big Steve. I got no further questions. No further questions about the heat shock proteins. Dr. Steve just set it straight for us. Yo, I gotta crack open a cold non-alcoholic beer for this one. Hold up. Come on. Okay. High concentrations of environmental ammonia can inhibit ammonia excretion in fish gills, leading to the accumulation of ammonia in tissues. In this study, acute ammonia exposure resulted in accumulation of tissue in the gut, liver, gills, and kidneys. I'm shocked. I'm shocked that ammonia exposure resulted in the accumulation of tissues in the gut, liver, and kidneys. You're killing the loaches. You're killing them. Exogenous taurine addition can decrease tissue ammonia concentration in large-scale loaches, suggesting that taurine increases the ammonia tolerance of fish. Man, the Red Bull is about to fly off the shelves. Fly off the shelves, man. Mick says, heat shock proteins are found in virtually all living organisms from bacteria to human. Wikipedia. Okay, guys, we're building the answer here as we speak. In fishes, GS catalyzes the synthesis of glutamine from glutamic acid and NH4 plus while glutamic acid is synthesized from A-ketoglutarate and NH4 plus catalyzed by GDH. Okay, this is getting too deep for me. Let me find the top line. Okay. This suggests that large-scale loaches could cope with high concentrations of ammonia through glutamine synthesis. Similar to the results of the study, ammonia-induced increases in fish GS activity has been observed in rainbow trout and yellow catfish. Taurine decreases ammonia accumulation in somatic tissues by improving mitochondrial function and regulating GS activity. In this study, injection of taurine during ammonia stress significantly increased GS activity in large-scale loaches. So I'll be honest, like one of the reasons this is interesting, so say I mean, this is like one paper, right? So like I'm not gonna start dumping taurine and everything, but I mean, one of the things when you're bringing someone that brings in a lot of new fish is dealing with fish that have ammonia related issues. So if ammonia and exposure to high concentrations of ammonia results in accumulating, you know, the ammonia in the tissues, you're putting them into fresh water. From what I'm reading, it sounds like they still got ammonia accumulation in them that their body has to process. So when I'm reading this, I'm thinking, is something like this helpful to if you're bringing in a bunch of fish, if it helps the body process this ammonia and get it out faster, could it help with bringing in fish that have a lot of ammonia damage and that you put them in fresh water, but they still got to deal with processing the ammonia that's accumulated in all these somatic tissues and stuff. That's something that I'm kind of like wondering as I read this. This study suggests that the study could further enhance glutamine synthesis to reduce ammonia in somatic tissues. Okay. In this study, liver GDH activity showed an increased trend in large-scale loaches under ammonia stress. This suggests that the amount of glutamic acid synthesized increases to bind to NH4 plus and convert it to glutamine. This is also consistent with a significant increase in liver GS activity. In contrast, taurine inhibited GDH activity in large-scale loaches exposed to ammonia, indicating that could help restore GDH levels under normal physiological conditions and alleviate the imbalance in amino acid metabolism caused by ammonia. Okay. AQPs are an inner membrane proteins that provide channels for water or glycerol in cell membranes. The physiological functions include the transport of water molecules, glycerol, urea, and gases. Previous studies have demonstrated that AQP3 and AQP8 can regulate the NH3 permeability of cell membranes to promote ammonia detoxification in fish. In this study, ammonia stress upregulated AQP3 and AQP8 expression in large-scale loaches, and this phenomenon has also been observed in Atlantic salmon, which makes sense since salmon would be another fish they're testing this kind of thing on, suggesting that fishes upregulate AQP's expression to increase cell membrane permeability to NH3 and thereby promote the excretion of intracellular ammonia. After taurine addition, there was no significant difference in AQP3 and AQP8 expression in large-scale loaches compared to the control group. In addition, the ammonia content in somatic tissues decreased. Excuse me. This indicates that taurine does not achieve ammonia tolerance through the expressions of aquaporins. RH glycoproteins are ubiquitous in bony fishes and are typical ammonia transporters. RH glycoproteins, ammonia transporters, okay? Yo, what's the fish fam link bot in the chat? Let's go. Oh my God. Okay, in this study, ammonia accumulation caused down regulation of large-scale gill and kidney transcription of RHAG and upregulated the transcription of RHBG and RHCG. So this indicates there's a significant difference in ammonia concentration between erythrocytes and plasma in high ammonia environment when the somatic tissue ammonia concentration is significantly increased. ammonia can directly diffuse from inside cells without requiring RHAG transport. However, when the environmental concentration is higher than the gill ammonia concentration, a fish must synthesize large amounts of RHBG and RHCG proteins to facilitate ammonia transport from the gill cells. Okay, let me see if I can understand this again. Okay, so in high ammonia environments, when the somatic tissue ammonia concentration is increased, the ammonia can directly diffuse from inside cells without the RHAGs transporting it. Okay. But when the environmental concentration, so the ammonia, in a high ammonia environment where there's ammonia concentration in the somatic tissues, that's part one. And part two is an environment where the environmental ammonia is higher than the gill ammonia concentration. All right. So in the second incidence, where the environment has more ammonia than the gill, the fish must synthesize large amounts of RHBG and RHCG proteins to facilitate ammonia transport from the gill cells. Okay. Sorry, just sometimes I gotta back up and just reread it and make sure I'm even taking a single bit of information from it. Exogenous taurine. I'm gonna look up the word exogenous. I mean, it seems pretty obvious, right? External. Exogenous taurine. Has anyone ever wrote that? Exogenous taurine. Okay, so like when it's like supplemented, it's not produced by the body. Exogenous taurine inhibited the upregulation of RHAG, RHBG, and RHCG genes following exposure to ammonia. This indicates that large-scale loaches do not require initiation of RH protein expression to promote ammonia detoxification under taurine treatment. In summary, this is going to be great. A summary of all this. Intraperitoneal injection of taurine decreased the content of somatic ammonia in tissues and upregulated GS and GDH activity in large-scale loaches exposed to ammonia. However, this had no significant effects on the expression of AQP or Rh protein-related genes. This suggests that taurine can promote glutamine synthesis in fish, therefore decreasing the in vivo ammonia content and increasing ammonia tolerance. So they're kind of going through like, okay, it's increasing ammonia tolerance. Let's eliminate the stuff that it is doing to help with that. So, okay, it's not affecting the expression of these AQPs or these Rh protein related genes that have to do with processing ammonia. You know, it's not doing this. Okay, but it is promoting glutamine synthesis, which is thereby decreasing the in vivo ammonia content, which is why it increases ammonia tolerance. Cool. Part of this study showed that ammonia stress can cause both significant immunostimulation and immunosuppression. At the same time, ammonia exposure also significantly affected the metabolism of large-scale loaches, resulting in the accumulation of ammonia in the tissues, increases in ammonia metabolism-related enzymatic activities, and alteration in the expression levels of ammonia transporter genes. So the ammonia transporter genes are those AQPs and the Rh glycoproteins, which they found the taurine did not affect. This suggests the glutamine synthesis is initiated and ammonia translators are upregulated to cope with elevated ammonia levels. After intraperitoneal injection of taurine, the ammonia concentration in the somatic tissues was significantly decreased in large-scale loaches, while gene expression of immune-related factors gradually recovered and glutamic acid and glutamine synthesis were further enhanced. Therefore, taurine can significantly increase GS and GDH activities in fish under ammonia stress to promote the conversion of ammonia and glutamic acid into non-toxic glutamine. All right, this is the sentence I'm going to try and remember. Okay, so the taurine significantly increases GS and GDH activities in fish under ammonia stress to promote the conversion of ammonia and glutamic acid into non-toxic glutamine. So if there is going to be a quiz on this, which I'm not saying there is or isn't, there's going to be another Kahoot quiz in the future. This would be probably what the question would be based off of. What is it? What is it that taurine increases to promote the conversion of what into what? and that's the conversion of ammonia and glutamic acid into non-toxic glutamine. These mechanisms enhance the tolerance to environmental ammonia and mitigate the immunosuppressive effects of ammonia stress in fish. Cool. And there's apparently no competing financial interests or personal relationships that could have appeared to influence the work in this paper. So guys, we made it through. This time we didn't find that the company promoting the product wrote it. Fantastic. Okay, this is great. I mean, I see Mick in the chat said the initial biochemical finding gave rise to a large number of studies of the induction of heat shock and its biological role. Heat shock proteins often function as chaperones in the refolding of proteins damaged by heat stress. Heat shock proteins have been found in all species examined from bacteria to humans, suggesting they evolved very early and have an important function. Thank you for that, Nick. Yeah, I guess one thing that this does for me is like when thinking about what makes a fish tolerant to ammonia, you know what I mean? Like, oh, this fish is hard. At the same time, ammonia. I forgot that my headphones were my microphone, so I took it off. I took it off and it turned the mic off. But I was just saying, like, when thinking about why something is tolerant to ammonia, I never have thought about, oh, is it because it's a very efficient at converting ammonia and glutamic acid into non-toxic glutamine? Like, that's never passed my mind. And, like, definitely getting ahead of myself because I haven't read that much about it. But I guess it's opening my mind to the idea of thinking more about what makes a fish tolerant to ammonia beyond just, oh, they can breathe air, from the environment. It can go to the surface and breathe air. Like, what are some of these other things? Oh, it's very good at converting this chemical into this chemical. And then with the taurine thing, like it seems like one of the things with it is something you could achieve through improved diet, you know? Like it kind of seems like the benefits that taurine are giving could be achieved through an improved diet. Like here, the WebMD, it says it's called an amino sulfonic acid. It occurs naturally in the body. The best food sources are meat, fish, and eggs. So it's something the body can produce. Probably has to eat something, decent to produce that. It has all these maybe positive effects. But when I see like the best food sources are meat, fish, and eggs, my mind like goes to like, why is the specific reasons live foods are so good for fish? You know what I mean? What are the nutrients that don't last as long when they're processed and cooked in higher heat and preserved in these other ways? And I feel like this kind of makes me think like, okay, this taurine thing, like what is the dietary item these fish could be eating that might contain some of these beneficial amino acids or whatever? And if it's found in like meats and eggs and all that, I'm thinking, yo, the live foods, man, right? Okay. I'm thinking about it. I'm thinking too hard. I'm thinking too much. Overthinking. Okay. Effects of taurine on ammonia and loaches. Perfect. Now let's get to the invasive loaches because I think that the idea of a loach invading. Dee Dee says it calms the nerval. Did it say it calms the nerval system? Uh, I don't know. I don't think I read that in here. I don't think I read that. The nerval system. I like how the word nerval sounds. Nerval system. If you're just joining, we're doing all loach papers for the most part for at least the first bit. If I keep going for a while, we'll get into some non-loach papers because kind of a backlog of stuff to go through. A lot of diet stuff. I'm really into diet stuff, and how it affects different outcomes and things. But just in general, just trying to look at some different loaches and how they fit in the world. We got a cave loach, we got a food loach. Now let's get into the invasive loach. Boom. Oh, that's not the one I was looking for. Whoops. Cam Jen said, I did see it said calming. Okay. Well, you have better eyes than me, Cam Jen. Validation achieved, Nick. Validation achieved. Okay, manage strictly necessary and confirmed choices. So here we have eDNA surveillance and population genomics of invasive loaches introduced to the United States. This was 2025 in August. Oh, I forgot to say that. A lot of these are all like from this year. I tried to do more papers that are just from this year because I find that to be I like the recent. There's a lot of good older stuff, but I like kind of being at the top of what the most recent things are. So August 7, 2025, this is from Mississippi State University, the Wildlife Fisheries and Aquaculture. Okay, this is a candidate for a degree of Master Science. So I think we're just like reading someone's paper that they did for school or something. I don't know. It's approved by a couple of professors and stuff. Let's see. Yeah, this is dissertations and theses. Okay, that's cool. That's a cool. Invasive loaches as your dissertation or thesis. That's kind of I can admire that. All right, let's see what they're up to. So the southeastern United States is an aquatic biodiversity hotspot, and the decline of native species is already facing extinction. Is attributed to the rapidly growing number of non-indigenous freshwater fishes. Next generation sequencing was used to analyze the population genomics of several populations of pond loach, Misgermis angulocaudatus. Let's look up this buddy. Let's look up this loach. The pond loach. Yep, we got a couple of or this seriously fish calls it the Oriental weather loach. Oh wait, is this a dojo loach? Is that what this is? Is this the invasion? Oh my God, guys, the dojo loach is invading. No, say it ain't so. So eating meat slash food with taurine helps calm the nervous system. Interesting. I mean, that's one conclusion. But see, Mick, you're starting to think like me. I think that even when it's something unrelated, like just reading some of the stuff in that last paper makes me think about, well, what else could what implications does this have elsewhere? And I think about, yeah, bringing in fish that have high ammonia damage, if it's not injecting them with taurine or people already probably feed them live foods and stuff, but there's probably variations of the things that would be useful for the conversion of ammonia into glutamine and stuff. And that probably something to look at. And I need to ask my friend in the Killy Club. He's a heart surgeon. Ought to know more. Ooh, okay. I like that. Zebra Aquatic says they're in a couple of creeks in New Jersey. So, wow. Here we got the whole map of the invasive dojo loach. I mean, it's not everywhere, but it's in a couple places. It's interesting how it's like East Coast, West Coast for the most part. Okay, three evolutionary significant units of this loach were found, suggesting invasion of multiple species in the United States. Additionally, non-invasive environmental DNA designed for early detection of invasive freshwater fishes, including M. angulocaudatus and the African jewelfish, Rubicachromis litoronexi, was developed. Oh, which fish is Rubicachromis Which jewel is this? Okay. Man, got all sorts of invasives. You should not always depend on one thing alone, so don't jump to conclusions too fast. For sure. For sure. But it does provoke thought. I barely understand any of the stuff I'm reading. So I definitely want to draw any conclusions off of me. Okay. So they're using eDNA, environmental DNA, to try and detect invasive species. Okay, well, I gotta look up. Well, they're probably gonna define eDNA. Oh, look, dedication. They dedicated this to their parents for their unwavering support through ups and downs of my academic journey, and for providing me with resources to learn and grow, and for patiently enduring my never-ending grass carp chatter, which is still ongoing. Jesus Christ. Yo, this is the grass carp. This is what the chatter's about, huh? On the kitchen table. Okay, he's thanking everyone. Or he or she actually didn't read, but shout out to the person that wrote this, for giving credit to the people that helped them. I want to see about this eDNA because that's kind of cool. Ben, you can't have a free fish tank today. I'm sorry. I'm sorry. Taurine, I gotta remember that word. Pick up a Red Bull can, man. All right. In the United States, introductions are a likely result of pet releases and aquaculture escapement. Escapement, I like that. From ornamental fish markets and goldfish farms. Within invasive systems, the dojo loach has been observed to increase turbidity and nitrogen levels while reducing populations of aquatic invertebrates essential to native species. No, they're definitely eating the isopods, man. Aquatic invertebrates are eating my isopods. A key factor in this fish's evasive success is its exceptional tolerance to harsh environmental conditions. Is that where it got the other name, the weather loach? Can endure desiccation and starvation for over 80 days. Bro. Surviving icing and temperatures exceeding 38 degrees Celsius, and persist in hypoxic conditions through facultative air breathing through the intestine. This species also exhibits high ammonia tolerance. Given the adaptability and popularity of the dojo loach among aquarium hobbyists, it's unsurprising to see this fish has established non-native populations in 22 U.S. states, from spring-fed streams in Alabama to the urban canals of Chicago, Illinois. Damn. Man, he already mentioned the freaking grass carp in here. He or she. All right. Let's continue here a little bit. So here's the tissue acquisition. So they got some tissue samples through extensive collaboration with multiple agencies and institutions. Samples included pectoral or caudal fin clips, depending on the acquisition sources, which were preserved in 100% ethanol and stored at -20 degrees Celsius until DNA extraction. All right. Whole genomic DNA was extracted from tissue samples using the DNeasy copyright blood and tissue kit trademark. Ooh, from a German manufacturer. Red Bull, you said? So is that what's calming the nerves when skiing down Mount Everest and such things? Of course, Mick. Of course. Come on. Okay, so we got the Dart Sec TM is also used for genotyping. It's a form of double digest restriction site associated DNA sequencing that combines next generation sequencing with Dart TM complexity reduction methods to create an SMP data matrix. Yeah, okay. Okay, SMPs were called population. Okay, let's get a little bit more into this. We don't need the details on how to use these machines. It's not going to happen. It's not going to happen at all. Oh, wait, did I reach the end? Oh man. Okay, population genetic analysis. All outgroups were excluded. Okay, okay. This is where learning to read would be very helpful. I think the whole paper might not be on here. Oh, results. There we go. Yeah. Loaches give you wings. Give the taurine to the loaches, and the loaches get wings. The 94 individual DNA sequences returned from DART-seq resulted in 85,000 variable SNPs and 40% missing data. All right. All right. Dataset was filtered. Where is the just general conclusion where I can understand? Oh, okay. I don't think we have the end of the paper. All right, I'm gonna have to pivot here. I didn't want to do this, but I didn't want to do this, but I'm gonna have to. I'm going to have to. Give me 1 second. There's more alien loaches. Don't worry. You thought there was just one? You thought I'd come that unprepared? Look, I can only look so far ahead. First record of the alien loach, Misc mohoidi, collected from Tokyo, Japan. Got a Japanese paper here, man. We got another freaking loach. Look at this. Look at this unit. In the Misc. It's got some more striping in a lot of these photos. I'm definitely noticing a trend with the loaches that are more the problem and how they present themselves. Let's see. Let's see. This is in Japanese with English abstract. Come on, man. Does anyone want to translate for me? So check this out. A female specimen of the snake weatherfish was collected from the pond of Arisagwa Nomiya Memorial Park, was identified based on having a total of 50 vertebrae, 20 caudal vertebrae, and the sequence of the mitochondrial DNA control region. This is the first field collection record of the species in Japan since the species' native range is centered around the Amur River Basin. Amur River Basin. This is in China. Yep, that's a little bit of ways from where they found it. It is presumed that the species has been abandoned after being imported for consumption or fishing bait. I can see how this could make good fishing bait. As a newly discovered alien species in Japan, a new standard Japanese name, Murahoso Dojo, is here proposed. Additionally, notes in taxonomy and identification are included. So this is where they got the invasive loach. This definitely looks like a very in the middle of a nice civilized area. Like I wouldn't expect to find an alien loach here. This is a photograph of the collection site. It's very nice, very pretty, very pretty. And here is the picture of the invading loach right here. A fresh specimen. I love the radiograph photos so you can see the bones. Look at that. Look at that. Yo, where is well, it's not a catfish, but still feather fins. I know he likes to scan photos of fish. That's kind of sick. These little spikes on the inside. Look, are these like the pharyngeal teeth or something? I don't know. It's like these little, is that just like the gills? I have no idea what I'm looking at. Mick said nice garden / park. Yep. It did look pretty nice. I mean, the loaches, they just get everywhere, man. You could just be in your local park, taking a walk, and then just see an alien loach right here. It's a crazy world that we live in. It really is a crazy world that we live in. Maybe we should all just go outside right now to our local parks and just see, I don't know. I don't know, like could there be an invasive loach in there that we've been missing out on this whole time? Just saying. What's up, Aaron Bone? Says hi, kids. How's it going? I don't think there's any kids in here unless Ben's still here. All right. Speaking of invasive loaches. Adaptation. Adaptation. So, adapt or perish. Adapt or perish. That's kind of like a daily question for myself. Barbara Jackson said, I wouldn't see anything at the park now. It's dark outside. Bro, come on. You got to get a headlamp. Come on, man. The technology exists. Aaron Bones says, if you're under 40 years old, you're a kid, then why don't they give me the early bird special? The discount, the senior discount. All right. If I'm not a kid, I want the old people benefits too. We're gonna have to like respect your elders. Like, where's my respect? You know what I mean? If I'm a kid, then I want I want the kid benefits. Like, I don't gotta do shit. No, I hear you. I hear you. But yeah, adapter pairs flexible habitat selection by an endangered and dispersal limited fish under variable flow conditions and invasive crayfish competition. Okay. Mediterranean freshwaters. See, when I think of Mediterranean, I do not think about freshwater for some reason. But Mediterranean freshwaters are renowned for their high level of fish endemism. I gotta look that word up. Endemism. The state of a species only being found in a single defined geographical location. Okay. I don't know if I've read that one. But at least 250 species are threatened with extinction due to climate change, biological invasions, habitat loss, and fragmentation. Our research focuses on the behavioral plasticity of dispersal limited fish as a mechanism to cope with human-driven environmental changes using endemic and endangered benthic species. As a model, the South Iberian spined loach, Cobitus holydyka. Oh man, hold up. This loach, this is the coolest looking noodle loach so far. It's kind of got like it's kind of almost got like a darter vibe going on with its pectoral, the way it's sitting on its pectorals and stuff. Like we're getting more in like the goby area with how it's sitting. Yeah, I'm rocking with this. I see okay, someone on the Loach forum got a group of them to keep. This is a thing. Okay. Excuse me. They collected a 7-year data set on microhabitat selection in a regulated river invaded by the exotic red swamp crayfish Procambris clarki. Ooh, isn't this from the United States? Yep. Yep. Sorry. Sorry. I'm up with one goofy cat in the fish room. You gotta have a goofy cat around in the fish room. It's the only way. They investigated habitat selection patterns under fluctuating flow scenarios and crayfish presence. This is what I was kind of more interested in this one, the whole flow scenario and crayfish thing, because I've got some crayfish. I'm trying to figure out where the hell to put them. They're cotton huge. Using a combination of univariate and multivariate analyses, our findings showed that C. palladica changed habitat selection patterns across years with differing flow conditions. The species selected deeper habitats with embedded coarser substrates and presence of aquatic vegetation under low flow conditions, but showed higher preferences for finer substrates with little embeddedness when flow increased. This behavioral flexibility may serve as a buffering mechanism against the increasing environmental degradation. However, our results also showed that C. palladica avoided positions with crayfish presence, indicating that the invasive P. clarki may be displacing the spined loach to suboptimal microhabitats, potentially through competitive exclusion. And the key words for this one is behavior plasticity, Cobitus palladica, endemic freshwater fish, flow disturbance, habitat selection, and Procambris clarki. Okay. The Mediterranean region is recognized as one of the 36 biodiversity hotspots of the planet. Oh, only 36, really. Ranking the second largest area with an exceeding 2 million km to 2. What, they ate Aspidoras, swallowed them in one piece? Wait, Barbara's goofy catfish ate Aspidoris. Is that goofy? That sounds murderous. Like I said, I wish I was under 40, but found out yesterday it was exactly 33 years since I visited a Metallica concert. No spelling error. Are you sure about that? Yo, your fish ate the Aspidoris and you're saying it's entertaining. It's crazy. Um, Unfortunately, yeah, the 250 endemic fishes are threatened with extinction, as we mentioned. Annual mean temperatures of both land and sea are now 1.5 Celsius above blah blah blah. Regional climate models project that the future warming will exceed the global annual mean by 20% and 50%. Alongside climate change, the main drivers of environmental change are now biological invasions. Oh man. Biological invasions and the anthropic alteration of river basins, particularly by hydropower projects that severely impact in-stream habitats. Dams may have an adverse effect on fish habitats through fragmentation of free-flowing rivers, alterations to flow and temperature regimes, reduced water quality, or changes to substrate composition caused by a decrease in sediment transport. Freyhoff et al. 2020 estimated that around 62% of critically endangered and vulnerable freshwater fish in the Mediterranean region have been highly impacted by currently operational dams and hydroelectric power stations. Man. Barbara said, I have no giant carnivorous catfish. Then what the heck did they do to the Aspidoris, man? Protecting Mediterranean threatened endemic fish requires the identification of their habitat preferences. We kind of need to do that too for some stuff. I started to think you got the Ipsibus gulper cat or similar. Barbara's got some red tails or something, some tomcats. I don't know what she got going on over there. For catfish was like featured on River Monsters or something. That's what it sounded like. Detailed knowledge of species habitat associations is essential to understand how anthropogenic habitat alterations could affect the suitability of in-stream habitats for freshwater species. So trying to figure out if they can adapt if they're displaced. Okay. Evidence from marine systems indicates that benthic fish species with highly specific substrate requirements are far less likely to shift their distributions in response to climate change compared to pelagic ones, whose distribution are primarily associated with dynamic variables like temperature or salinity. There's no such clear evidence for freshwater benthic fish, but it's reasonable to think that their strong affinity for substrate composition can retain the benthic species in areas with deteriorating environmental conditions. So if they're building a dam or something else is getting jacked up where they're living, maybe they like the substrate too much. They don't want to leave. They're going to hang out and they're going to get absolutely demolished. Yeah, that'd be unfortunate. Does the interplay of limited mobility and strong substrate affinity may increase the risk of their disappearance? All right. All right. Let's go down to where this starts. Materials and methods. So the study was conducted in the River Balac at El Torno, Ciudad Real, Guadana River Basin, and then just Spain. Spain with some coordinates at 600 meters above sea level. Okay. The main environmental impacts in the study are those derived from water withdrawal for irrigation and river fragmentation. The Gardena River Basin is one of the most complex river basins in Europe. It's prone to water scarcity. There's strong competition for water among stakeholders, and surface and groundwater bodies are highly regulated and overexploited. Overexploited? No, say it ain't so. Say it ain't so. The Gardena River Basin has a Mediterranean continental climate with a distinct dry season and marked seasonal fluctuations in temperature, which involve extreme temperatures and limited precipitation during summer, and that means severe low water levels. And this is the mean monthly flow in the river during the study period. Wow. This is some cool data, honestly. The river bullocks. Still going, still going. Look at this freaking flow history, man. What happened here? What happened in 2004? Crazy. Okay. We got some detailed data going on here. Three flow categories were established based on the daily flow data recorded on the 5 weeks before the fish and habitat sampling period, April through May. Previous studies have evidenced the importance for accounting for the short-term antecedent flow to improve the performance of ecological models relating flow conditions to biological processes. All right. All right. All right. The native fish community of the study site is well preserved and consists of the Iberian small-headed barbel, Luciobarbus microphallus. All right, little barbel. Oh, not little. Maybe not little. Holy. Worm during summer. The Iberian arched malthanace. Okay. All right. Yep. It's like a cool little fish. Iberian arched. We got a little display photo here. Hold up. They put this damn thing in a bottle. There's nice little sparkles on top. I like that. That's a cool little silvery fish. Yeah. Southern straight malthanace. Okay. Pseudocondystroma wilcomi. Let's see. Okay, this one's got some yellow fins on it though. It's a little bit cooler looking. It's got some sparkle though. Caladino squaleus albraconoides. Looks like a mini trout kind of. Oh, here we go. Here we go, mini trout. May as well say it's a CPD. Here we go. $10 a fish. This one looks a little bit weirder. It's got a cool like turquoise sheen in some of these photos. So all the watermarked ones at least. The southern Iberian chub, Scoleus tartasicus. Scoleus tartasicus. These kind of all look the same. I'm not gonna lie. Okay, here we go. This in the southern Iberian spined loach, which we looked at, the C. palladica, C. palladisa, C. palladisa, however you want to say it. I think it's kind of a cool looking loach. Like I said, it's kind of how it sits in some of these photos. It's reminding me more of like the darters and gobies kind of a vibe because it's got that silly face. So heat shock proteins created inside those fish, making them tolerate the bad waters. Listen, Nick, you're reaching beyond my ability to ponder and theorize. I just learned they existed. Okay, non-native fish are absent, but invasive exotic red swamp crayfish from the United States, Crambus clarki, is widespread and abundant. Jesus. Oh man. Okay, to model the spined loach habitat selection, microhabitat availability and use were characterized in a reach of about 120 meters long and 32.8 plus whatever 3.6 meters width. The study was sampled once a year from 2003 to 2008, and in 2019, always on the same dates, the 5th or 6th of May. These dates mark the onset of the dry hydro period when river flow allows sampling throughout the reach and temperatures are not yet very high. In later months, low flow and high temperatures are stressful for the spined loach, especially for young of the year individuals. At such time, fish sampling is inadvisable. During autumn and winter, flow is too high for sampling. Therefore, the spring offered us the best opportunity. They were sampled by electrofishing. What the hell is electrofishing? Oh, they're just shocking the fish? Oh, this is hacking. This is hacking, bro. They got the metal detector in there with the fish. Bro, what the hell? This is cheating. Electrofishing. Yo, they're gonna have the AI robots out here looking like this, man. Looking like this. Yo, what the kind of battery he got tied to his back? This looks hazardous for everyone involved. I don't think fishing should be in the name of this. Electrofishing, there's nothing fishing about this. I mean, there's something fishy about it. Can you imagine seeing someone with this get up walking around near you? Don't get out. Getting them used to the repetition of these sudden shifting conditions. Makes sense. Okay. So they're electrofish captured, weighed, measured, and returned alive to the river. Oh, okay. Well, that's nice. That is very nice. All the work complied with the current Spanish and European conservation legislation and strictly adhere to the regulations on the handling of wild animals. Okay. So they're just saying we didn't do this anything illegally. They've been doing this for years. I've seen them have giant outriggers. Yeah, that's crazy. This electrofishing thing, like I said, I just feel like it's so unfair to the fish, man. Give them a chance. Okay, where they captured a loach, water depth and distance to the nearest bank were measured in the exact point where the fish were captured. You never heard of that? My father-in-law saw his neighbor do that in Florida with a car battery. His neighbor returned them to the frying pan. No, Barbara, I don't know anyone that's using car batteries to shock fish so they can eat them. That's just not something I've come across. How else do you sample the populations? I don't know, do something that takes skill. You know, like catch them with your bare hands or like your mouth, like in your teeth or something like that. You know? Think about it. Wherever, okay, so they measured stuff where they caught it, the proportion of aquatic vegetation, substrate composition. Okay, now we're talking an embeddedness of the substrate. How do you even measure that? Okay, percent of the substrate that was covered in silt. That's the embeddedness of the substrate. Zebra said it also works well for removing invasive snakeheads. Oh, okay. Barbara said it was a long time, like 65 years ago, or so ago. Yeah, I don't think I would have been able to witness that. Not quite. What the heck does science skill to do with science? We're not sport fishing. Why not? Why shouldn't science also require skill, Steve? You don't want our scientists to be able to grab fish with their bare hands? I don't understand why you're fighting me on this. I just want to demand better. Next, we have some rare endemic loach fish close to the Stockholm area too, if I remember. Annual controlled counting of the population in the creek here. Okay. Like what? Should I be shocking my fish to catch them now? Is that what we should be doing? Unloading the camera, the car battery into the low boys here. Substrate composition or particle size composition of the substrate was described following the classification criteria established in this paper from 1983. Okay. To determine microhabit availability, the same variables were measured with the fish samplings at randomly selected points throughout the study site. 5 to 7, depending on the channel width, random positions per 10 meters of the river length were assessed. Selected random positions varied across sampling surveys. They used two different approaches to evaluate habitat selection. One, univariate analysis to compare availability and use for each measured habitat variable. And two, multivariate analysis to account for nonlinear and interactive effects of habitat variables. So for the first one, they check if the habitat data met the assumption of normality of distributions using the Shapiro-Wilk test. Most variables violated the normality assumption, so they were transformed using the natural logarithm and the non-percentage variables and the arcene of the square root of the variable and the percentage variables. Yo, if anyone actually understands what I just read, yo, hop in the chat, man. This is crazy. For each habitat variable, depth, distance to nearest bank, aquatic vegetation, substrate category, substrate embeddedness, we tested whether availability differed among flow categories and the use by the spined loach, if the use by the spined loach differed among those flow categories, and 3, availability and use differed within each flow category. Okay, let's go. Let's go to the results. We've got quite a lot of buildup going on here, man. Okay, depth availability in the study varied between 5 and 116 centimeters throughout the study period. Availability of aquatic vegetation was relatively low across years. Okay, over the study years, sampled quadrats were dominated by coarse substrate, like 59%, and gravel, 31%. Fine substrate was scarce, 9%. The level of substrate embeddedness was usually high, 57%. All microhabitat variables showed significant differences among flow categories. Let's look at, okay, so we got the different flow categories here. I accidentally clicked the thing that made me lose my place. Gravel hides eggs well? Yeah, man. I'm telling you, man. Rock piles and gravel. Rock piles and gravel. Steve says the Maryland DNR uses this electro technique as well. Disgusting. We got hackers in the state. The habitat use data was recorded for a total of 240 loached individuals, ranging 30 to 115 millimeters in total length. Age structure was clearly skewed, although individuals up to 8 years old were found. Significant differences were not found in the use of any habitat variable between age classes. Consequently, age did not significantly influence habitat selection under any flow condition, so all habitat use data was pooled together. JP Jeffries says, maybe I could use electroshock on my cichlids, teach them to get along. Jesus. This isn't the way of opening your mind. This kind of stuff is supposed to be doing. Habitat used by the spine loach differed significantly among flow categories. Under low flow conditions, the spine loach occupied positions with medium depth, dominated by coarse substrates, and with high proportion of aquatic vegetation and embeddedness. So lower flow, coarse substrates, more vegetation. Under medium flow, the spine loach occupied a similar microhabitat than under LF, but with a higher proportion of fine substrates and much less aquatic vegetation. Under high flow conditions, the occupied positions were deeper and further away from the bank. The substrate were dominated by gravel and coarse elements, and the percentage of embeddedness was significantly lower than under the other flow categories. Something I'm thinking about as I'm reading this and some of the other papers I've read is there is something about how some of these fish that we think of like being high flow fish, some of them are only in high, medium, these different flow categories for certain parts of the year. You know what I mean? Now this general one, I'm not sure if their change in high, low flow, whatever, is just by choice or just that's what they're faced with. But yeah, just interesting. Different flows, different parts of the year, and then these different flow areas have different kinds of habitats. I would imagine if a low charge was used, the fish probably don't have damage. Sounds like they wake up okay. This is like the new fish catching method. Jesus. Okay. So under high flow conditions, yes, the occupied positions were deeper and further away from the bank. The substrate was dominated by gravel and coarse elements, and the percentage of embeddedness was significantly lower than the other flow conditions. Univariate analysis showed that the spine loach selected or avoided only certain habitat features and that selection patterns differed across flow conditions. Oh, that's interesting. So they have preferences, but those preferences change depending on what the flow was. Under low flow and medium flow, the spine loach avoided positions dominated by gravel and showed a marked preference for those dominated by coarse substrates with an elevated percentage of sediment. Whereas at high flow, it reversed the pattern, selecting gravel and avoiding coarse substrates. It's also significant that under medium flow, the spined loach avoided the deepest areas and differentially selected the scarce microhabitats with a high proportion of fine substrates. Under high flow, the spined loach selected positions located far from the banks. For the rest of the variables, the habitat use followed the dynamics of habitat availability. when considering microhabitat selection as a nonlinear multivariate process and accounting for the presence of crayfish, the most parsimonious models showed a major role of the invasive crayfish in the microhabitat selection of the spined loach. Oh man. Regardless of flow conditions, the spined loach was mainly present in crayfish-free areas. However, some habitat features were important under different flow conditions. Under low flow, depth and aquatic vegetation appeared to be relevant variables for spined loach, which preferred deep microhabitats with abundant aquatic vegetation, and that is again in low flow. Presence under medium flow was mainly explained by high levels of embeddedness. So that's the amount of silt over the substrate. So high levels of embeddedness. And under high flow, it was more likely to find the spine loach far from the banks in fine and gravel substrates. Age didn't have a significant effect on habitation in any of the models tested. So here we have spined hill loach presence and the presence of the crayfish. So, and against the flow. Okay. Model showed nonlinear relationships with some explanatory variables with effective degrees of freedom. Okay. Our findings demonstrated that the southern Iberian spined loach exhibited different habitat use and selection patterns under contrasting flow conditions, a result evident in both univariate and multivariate analysis. Chloroform more like borophyll? Bruh. Oh, here we got a chart that I can't read, but this is some of the data with the crayfish versus loach. And all the this is kind of complicated, all the different variables they have, man. It's kind of wild. And they did it for a bunch of years. Like, this is a cool study. Okay, multivariate analysis underscored the dominant role of exotic crayfish presence in influencing habitat selection. Additionally, the spined loach showed preference for deeper areas with aquatic vegetation under low flow conditions, interstitial sediment under medium flow, and fine and gravel substrates under high flow. Other burrowing species typically prefer fine substrates, mainly sand, also mud and clay, combined with patchy, dense microphytes for predator protection. This substrate composition is the most suitable considering the feeding behavior of the spined loach, which pumps fine material through its mouth and extracts food with mucus. Gross. This general pattern for Cobitus species is consistent with the habitat presence of C. palladica preserved in the river Balak under medium and high flow. Sounds like crayfish boil parties are in order, Barbara says. Yes, indeed. Okay, contrary to previous studies, the spined loach and the river bollock selected coarse substrates highly embedded by sediments under low flow conditions. Several studies show that other small benthic fish, such as sculpins, darters, and gobies, often selected coarse substrates that provide profitable feeding positions. Yo, next time I'm lounging on the couch, in the bed, eating ice cream or some other snack like a pig, I'm gonna call that a profitable feeding position. Okay, the core substrates provide profitable feeding positions or shelter from predators. Okay, besides, interstitial spaces between cobbles are essential habitats for benthic fish and some loach species, as they use them for refuge. In habitats dominated by coarser substrates, the relevance of muddy sediment for spined loaches increased, which is consistent with our findings for C. palladica and the river Balak. Perhaps the key point is that the sediment surface provides a good food supply, potentially offsetting the species' typical intolerance for this anoxic substrate. A relevant question is whether C. palladica is freely selecting microhabitat conditions or is seriously constrained by the presence of the invasive P. clarki. Literature shows that non-native crayfish and native benthic fish often overlap in their niches, suggesting potential competition for safe and profitable microhabitats in the river Balak. Muhammad et al. In 2023 experimentally demonstrated active competition for shelter between invasive crayfish and signal crayfish in a native stone loach and bullhead. Okay, so the crayfish seem to really be messing up the habitat selection over here, man. To date, no study has measured the specific impacts of the red swab crayfish on endangered Cobides species. However, previous research has shown that small benthic fish are particularly vulnerable to crayfish invasions, with strong evidence of a recruitment failure under natural conditions. Some experimental works have evidence that invasive crayfish species can significantly alter substrate composition, particularly through gravel movement and the removal of fine sediments. Indeed, bioturbation caused by the burrowing behavior of P. clarkii has been observed in the River Gardena near the study area, leading to major consequences in water quality, including an increase of dissolved inorganic nutrients, turbidity, and hypoxia, excuse me, as well as reduced available organic material for benthic macrofauna. Man, the crayfish are just jacking this everything up, man. It's like the list of things that they affect negatively, it just keeps going on, man. Hypoxia, turbidity, dissolved inorganic nutrients. Oh my. With our data, we cannot attribute the macrophytes shortage in the area solely to invasive crayfish, as an abundance of submerged vegetation also depends on the flow regime. But P. clarkii could be contributing to the decline of this research. So in conclusion, they found that this loach can select different microhabitats depending on the interannual variation in flow rate, which gives them some resilience in the face of increasing environmental degradation caused by the combined effect of climate change and flow regulation. The presence of spined loach in good numbers, coupled with a well-preserved population structure over time in the study reach, suggests this species can tolerate suboptimal microhabitats such as coarser species embedded in the sediment. However, additional such stressors like the crayfish are a risk because they push them into suboptimal habitats through competitive exclusion, potentially. Wow. Wow. A dispersal limited species of conservation concern action should prioritize removing invasive competitors such as crayfish, preserving suitable substrates, and improving connectivity among habitat patches to ensure natural gene flow, thus maintaining genetic diversity, which enables populations to adapt to environmental change. Amazing. And this was funded, open access funding provided thanks to Crew SIC agreement with Springer and Nature. No competing interests or financial interests or personal relationships to influence the work. Perfect. And this is licensed under Creative Commons Attribution 4.0. Let's go. Roaches of the sea/water crayfish. Mix it. Yeah. How you guys feeling about that? That was kind of interesting. I feel like any paper that goes in depth about the habitat selection of any fish is relevant when thinking about keeping fish, you know? It doesn't necessarily have to be the fish you're keeping, but just, it gives me a lot to think about how a fish interacts with its environment in the wild and chooses where it lives. I never really thought about flow rate, not flow rate preference, but like, okay, if I'm in high flow, I want to be in the coarse substrate versus the sandy substrate, or whatever. So it's just some things to think about. Let me put the link to this in the chat though. I like this one. This was good. I could read most of it. Or I could read most of it. Let me see. How we feeling? You guys feeling all right? How long have we been streaming? We're almost to 2 hours. We're almost in the green zone. Under 2 hours. It's like, what are we even doing? What are you guys feeling? Want some disease stuff, some diet stuff? I'm thinking. I'm looking at my list. What do I feel like reading? What do I feel like reading? There's one paper that's about like how to preserve the loaches for food. Uh, JP Jeffrey said, "Good loach deep dive. Thank you. Any new loach species in the hobby?" I didn't have any ones that were hobby specific on my list for this one. There was one kind of interesting Okay, let's check this out. The Loach nerds might appreciate this, but I don't know if I'm gonna be able to read it. So we're about to find out. But check this out. So, first evidence of secondary sexual dimorphism in the freshwater fish family Botidae, a newly recognized sinomorphy of loaches. Whew. Okay, I told you I'm all about the sexual dimorphism, man. So within the freshwater order Cryptoforms, loaches from a nonophyletic lineage comprise 9 families with more than 1,400 species. Secondary sexual dimorphism is widespread among loach families, most notably in the form of enlarged pectoral fins, tubercles, or other ossified structures on the pectoral fin in adult males. To date, the family Lotidae, phylogenetically sister to all other loach families, were reported to lack such sexually dimorphic structures, leading to the hypothesis that the common ancestor of loaches did not exhibit sexual dimorphism. Wow, is the telling the difference between male and female loaches were that bad? They were just like, yeah, the common ancestor. There must have been no difference. But here we report the presence of sexual dimorphism in 8 species of Botidae, Leptobotida delicata. Let's see. Man, this got some crazy looking. It's a lizard, bruh. It is a lizard. Dimorphed. Great name for a rock band. L. gillianensis. Okay. I'm not going to read every single one, man. L. microphalma, L. tenops, L. tishingi, Parabotia fasciatus, Cenobotia pulchra, and S. robusta. In all species, adult males possess longer pectoral fins than females. Additionally, males of L. gillianensis and L. tichangi exhibit larger pelvic fins, while males of L. microphthalma have larger anal fins, and L. bellicata, L. microphthalma, and L. tichangi portions of the dorsal surface of the pectoral fin bear rows of tubercles. The 3 genera displaying sexual dimorphism belong to 2 different subfamilies, demonstrating that sexual dimorphism is widespread across Botidae and not restricted to a single genus or subfamily. Our results show that sexual dimorphism is present in most of the basal family of loaches, suggesting that it represents a sinomorphy of loach fishes. Holy. Okay, here are some of the loaches they looked at the sexual dimorphism of. Feast your eyes. My eyes are feasting. Okay, pictures not to scale. Thanks for that. Okay, let's see. How did they go about this? Okay, we got a table there. That's going to be a lot for me to try and read off the cuff. Okay, that's a lot. Okay, in total, 19 species were examined for the presence of sexual dimorphism. The sex was determined by gonadal inspection, and only species with developed or developing gonads were included in analysis. That's nice that they checked that. What's up, Aman Shashidiz FSH? I tried. How's it going? Okay, visual inspection and photographic documentation of morphological details were conducted using an Olympus SZX7 stereo microscope. Yo, how much is this? Let's see. Oh, this one's only 2,000. This one's 3,000. Light work, man. Light work. Yeah. Yeah, you guys got this. I'm sure you got this at home. Equipped with a UI camera. All measurements and counts follow this individual's work. Standard length and 26 morphometric characters and 5 fin counts were measured point to point using digital calipers to the nearest 0.1 millimeter in a subsample of each species. From these measurements, characters indicative of sexual dimorphism was selected. Standard length and dimorphic characters were subsequently measured for all species, and for each sexed adult specimen, relative fin measurements were calculated. These values were visualized as scatter plots using ggplot2. I'm sorry. GG plot 2. Bro. Wow. GG plot 2. Okay. Yeah, this looks crazy nerdy. This looks fantastic. I mean, look at the logo. We got data points. Fantastic. GitHub. Let's go. Pigmentation patterns on body, head, and fins, as well as shape of genitalia papillae, were compared between males and females by visual inspection using the above-mentioned stereo microscope. Okay. So let's take a look at these dimorphic loaches. In all 8 species, males exhibited longer pectoral fins than females, a pattern present in species with relatively small fins, as well as those with relatively large fins. Additionally, males of L. gillianensis and L. tshangi had larger pelvic fins than females, and males of L. microphthalma had larger anal fins. So here we see males and females side by side. It looks like the left is the male on all these, and the right is the female. So looking here, there's a pretty big difference in the pectoral fin size that I think we could probably tell by looking at it here. For A, like I see that seems pretty clear. That does seem pretty clear. B, okay, I can see B too. Maybe it's just the angle they took the photo at, but it's not as obvious. C looks way close. Like, I don't think that's obvious enough for me to be able to tell, especially if they weren't both like grown to around the same size. D also I think would be hard to tell with them in an aquarium. Like the what with A, I feel like I could tell if I saw that fish in an aquarium and I was looking at the size of the pectoral fins, like I could see those are, from the side, those are clearly different. E, I guess, is pretty obvious. You can see here the one on the left, male again. All the ones on the left are males. All the ones on the right are females. This one, the male's head looks a little bit crazier, honestly. F, I guess nah, that one looks like, I think I would have a hard time. G looks impossible. I don't think I could do that. There's a little bit of shape difference, but from the side, I don't think I could tell the difference between that. That's too close. Same with H. H also looks hard, but I can see that there's a difference. I just, in an aquarium, would I be able to tell? I don't know. Here we got a chart with sexual dimorphism in fin size. So on the vertical axes, we have pectoral fin length, and then on the horizontal is the species. And then it looks like the different colors should be male and female. No, maybe not. I feel like that's what it should be. Yeah, males in purple, females in orange. Okay, so for each species here, we can see the length of the pectoral fins on a chart. Robert said, good thing the fish can tell the difference. I mean, that's a big assumption. So if I felt like spending the time, we could go back and see like if I'm right about which ones were obvious or not. But here you can see some of them, the pectoral fin length is a lot closer than others. Like in Taniops, I feel like there's some that's pretty close. L. bellicata seems pretty far apart. L. gillianensis seems pretty far apart. So let's just check what's A. A was seen pretty. Yep, A is L bellicata, and that one seemed pretty obvious to me. B is gillianensis. B doesn't look that obvious from the photo, but in the chart it looks like they're for the most part far apart. Okay, microphthalma. They say this one's pretty far apart. I guess that photo isn't the worst. But okay, this is a cool. I can read this chart. I can read this chart. I appreciate it. We have some pelvic fin differences here with L. gillianensis and L. tshangi. Very cool. Very cool. Oh, we got some close up. Most analyzed Botiidae exhibited radial skin folds on the dorsal side of pectoral fins of both sexes. We refer to them as dorsal fin ridges. These ridges were composed from a skin fold filled by most likely fat cells and appeared darker and thicker than the fin membrane. They're anchored along the dorso-posterior margin of the most pectoral fin ray. Dorsal fin ridges occurred along the last two unbranched and first 5 to 7 branched rays, decreasing in thickness toward posterior rays. Their size and prominence varied, increasing with specimen size regardless of sex. Well-developed ridges along most of the fin ray length, broadest at mid-length, and sometimes covered in the interspace between the adjacent rays. In L. bellicata, the ridges were broad, flat, and oriented dorsal-posteriorly, similar as in the Nemachielid genus, Chernomochilis. What's up, Beto? How's it going? Resembling automotive spoilers. It is likely that they have a hydrodynamic function. Yo, Featherfin, this is for you, buddy. Resembling automotive spoilers. This paper is comparing the fins to race cars. You did it first, man. I'm giving you the credit, but I just want to say it made it into the paper. Ah, okay. Let's take a look at this. These ridges. This looks kind of gnarly up close, I'm not gonna lie. So, presence of tubercles on pectoral fins of adult males of sexual dimorphism in 3 species. Left pectoral fin and dorsal view, right enlargement. So, if you're looking for a way to tell if a fish is a male or not, see, when people say tubercles on the head or whatever, I always think of them being a little bit bigger. You know what I mean? Like, do I gotta zoom in on my fish's fucking pectoral fin and then look at the damn, like these damn little dots? This is what I'm supposed to be seeing with my eyes? Jesus. Like, you can't even see them in the unenlarged thing on the side. I mean, A, you can kind of see it, but the rest of them, they had to zoom in. So the tubercles were present in these 3 species on the dorsal surface of the pectoral fins, but absent in females. Okay, guys, you try and tell the difference between these hillstream loach sexes? Like, maybe we need to look closer for these tubercles. Mick said, when watching and hearing this, we can see how much research is still needed in this world. The more we learn, the more we want to keep researching. I said, yeah, man, that's why I like doing these streams, because I don't really understand or know any of this stuff, but it does get you thinking. And just so you guys know, I'm staying up to date on shit. This was published December 16th, 2025. Come on. Come on. I'm on it. I cataloged immediately. Right onto the stream. Let's go. Okay, tubercles were associated with the last 2 unbranched rays and the first 4 to 7 branched rays. They had a round base, conical lateral profile, and pointed tips, sometimes sharp, sometimes blunt. In L. microphthalma, tubercles were fewer, up to 500 per fin, and smaller, often confined to the midline of rays. In L. pellicata and L. shangi, tubercles were more numerous, up to several thousand per fin, and larger, with distinct distribution patterns on the dorsal surface of the 2 unbranched and first branch rays, and on the dorsal surface of dorsal fin ridges and subsequent rays. Their abundant size and distribution varied among specimens, generally increasing with male size and gonadal development. In L. shangi, 2 small males with only slightly developed gonads lacked tubercles, whereas larger males with mature gonads displayed numerous tubercles. A different type of tuberculation was observed in some S. pulchra and L. tenops specimens, consisting of numerous very small tubercles on the dorsal parts of head and body. However, these tubercles occurred equally in males, females, and juveniles and do not represent a sexual dimorphism. Great, great. So the ones that are easier to see that aren't helpful. Mick said, new, really, Nick? It's 2 days old. Are you for real, Nick? Shut up. Until now, no sexual dimorphism has been reported in Botidae. The present study demonstrates that Botidae exhibit morphometric and structural sexual dimorphism manifested as enlarged pectoral, pelvic, and anal fins, as well as tubercles on the dorsal surface of the pectoral fins. Okay. During investigation in the study, enlarged pectoral fins of males were also noted in additional species, Botia striata, Cinnabotia longiventralis, S. superciliaris, Ambastia sithiamuki, and A. nigrolineata. But due to the low number of available ripe specimens, This species was not included in the present data set. Okay, further investigations are likely to reveal additional instances of sexual dimorphism in other Botidae species in genera. In contrast, tubercles on the dorsal surface of pectoral fins were observed exclusively in species of the genus Leptobotia. Further investigations of more species will show if it is a specific trait of this genus or an artifact of the limited materials in the present study. Nick said, JK, Nick, you're awesome, buddy. The damage is already done, Nick. You can't unring that bell. Nowbat 35 previously assumed that Botidae lacked any sexual dimorphism, using this premise to postulate that the common ancestor of loaches did not exhibit sexual dimorphism. Our results demonstrate that this assumption was incorrect, mapping the known instances of sexual dimorphism in loaches onto a cladogram derived from molecular phylogenetic studies. That shows that pectoral fin sexual dimorphism occurs across multiple loach families, including the Botidae. Therefore, we postulate that the presence of the sexual dimorphism of the pectoral fins is a shared character of loaches, and that a common ancestor likely possessed enlarged pectoral fins in males, potentially accompanied by tubercles on the first branch fin rays. Here's all the families with the male characteristics, male specific characteristic states. And Botidae, it has both enlarged pectoral fin in males and tubercles on the fin. Cyprinidae, Catostomidea, and Geniochilidae. They all have tubercles on the head in males. Then Valentilidae, they have the enlarged pectoral fins. Cobitidae, they have the tubercles on the pectoral fin in males. Nematochilidae, they have both the tubercles and enlarged pectoral fins. Then Gastromyzon today, they have the tubercles on the pectoral fins in the males. Yeah, I need to get some gastromyzons, man. Where they at? Where are they? Hold up. Okay. Anything else they got to say about these sexually dimorphic beasts? Oh man. Okay. Conclusion. Yeah, they just concluded, hey, you said it didn't exist. We've shown the dimorphism. Suck it. That's basically what they're saying. Man. Yeah, that was actually a more useful one. I'm about to take a look at some loaches the next time I get them. See if I can get a close-up photo of the pectoral. One second, one second, one second, one second. Okay. How you guys hanging in there, man? You ready to go to sleep yet? Come on. I know you're getting tired. Barbara said that was rather interesting. Yeah, no, I like that. That was interesting. I like that one. Should we stay on loaches or should we branch out a bit? I kind of feel I'm feeling like branching out slightly. I know just because that one I feel like was so satisfying. That one was pretty satisfying. I'm not gonna lie. And I kind of have one. I kind of have a couple uh immune system and otherwise related that I'm kind of interested in. Oh, but this one isn't recent enough. We got to do only ones published recently. I've got it strict. Nick is in here branching out. All right. Let me see. Oh, you know what? There's one more Loach one. It's 7:40 p.m. here. The night is young, dog. You get an early sleep. There's one more Loach related one, but it's like it's not just Loaches, so it's branching out a little bit technically. Um, and I think it'll be generally interesting. Gonna order some aquahuna soon. Oh, then you might need this one. Wally. If I can read this. Oh man, this whole thing is in Russian. Never mind. I forgot that one was in Russian. You win. You win, Johnny. You win, Johnny. We'll branch out a little bit. We will. Except, will we? Hold up. Hold up. 4:40 a.m. here tonight. Go to sleep, Mick. Many different loach species you can look up. Yeah, there's quite a lot. Okay. Last loach one. Last loach one. Okay. But it's diet related, so this is to investigate the mechanism by which alanine glutamine alleviates intestinal damage in the loach, Paramysgyrnis gaburinus. And isn't this the dojo loach or something that we did earlier, right? Or is this the one they be eating? No, this is the one they be eating, the large scale loach. Okay, so this is kind of crazy. So alanine glutamine, let's just start. They're probably going to define it, but I'm just going to see. Wow, Thermo Fisher sells this stuff. A synthesized dipeptide, which can be digested to form stable L-glutamine in vitro. Ooh. The only loaches I have are dwarf zebra, Vietnam loaches, micro Nemeschalis cruciatus. They really put a Harry Potter spell as the loach name. Hey, these are cool though. Yeah, these are cool. Okay, so they're investigating what mechanism lets this allergen alleviate intestinal damage in the loach. They fed the fish diets supplemented with different levels of allergen, and then challenged the loaches with Aeronomas hydrophila to induce enteritis. Jesus Christ. Oh my gosh. This is, these papers, it's like, did this hurt the fish? Is there a specific loach you have your eyes on? I do. What is it? I do. It's the green one. I can't remember the name of it right now. I'm blanking. But we should look at the one that I'm interested in. Actually, there's some loach species that we should actually look up after this. Actually, some hill streams. See if we can find some papers on the fly. So here's the experiment. We got a great graphic showing the food with the percentage of the supplement they're putting in there. So AG1, AG2, AG3, AG4 has different percentages from 0 to 1.25%. So then here they show injecting the loaches with Aeromonas hydrophila to give them this infection in the intestines. Uh, then they sample the intestines at 0 hours, 6 hours, 12 hours, and 24 hours. They look at the infection, some expressions, and the mechanism of the inflammatory response. So we have the innate immune cells, the macrophage, multiple cell cypros, damaged tissue, intestinal inflammatory response, and repair of damaged tissue. All right, let's see how the fish, did they kill these damn fish? It's gonna be one of those. Okay, glutamine is a conditionally essential amino acid in animals that plays vital roles in protein synthesis, energy metabolism regulation, growth promotion, and immune enhancement through supporting antibody production, thereby strengthening overall disease resistance. While endogenous glycan synthesis meets metabolic needs under normal physiological conditions, the production becomes insufficient during stress. See, man, it all comes back to stress. Robert asks, what's your favorite loach, Johnny? That's a good question. So this is interesting. It says its production is insufficient during the stress, so the body can make it, but when stressed, it can't. And that is necessitating dietary supplementation to maintain homeostasis and reduce stress-induced metabolic disorder and tissue damage. Like, I'm telling you, this stuff really makes you think about what the specific functions of, okay, different ways mistreating your fish can kill them, I guess. Okay. However, the use of free GLN in animal feed is hampered by its poor solubility, low thermal stability, and degradation into toxic pyroglutamic acid and ammonia during processing or storage. In comparison, alanyl glutamine, a peptide composed of alanine and glutamine, shows superior solubility and thermal stability. It is efficiently hydrolyzed in vivo to release GLN, which is observed through the intestinal mucosa, offering a more effective and stable means of GLN delivery to sustain physiological functions. Okay, so the free GLN itself, not that great as an ingredient. This alanine glutamine contraption is and delivers the GLN as it's hydrolyzed in vivo. It releases the GLN where it can then be absorbed through the intestinal mucosa. Dope. An effective and stable means of GLN delivery to sustain physiological functions. In aquaculture, fish health and growth performance are crucial factors influencing, can you guess, economic outcomes. Robert Jackson said another argument for live food. That's what. So I think it's known like live foods are good, and it's probably known that it's because of these nutrients that are harder to put in certain foods. But I think what this kind of stuff starts to make me want to do a picture of is okay. Like for instance, some of the papers we've read on Daphnia and Boina show that they're more complete and a better food than Artemia. So better understanding the things that make that so, but Artemia lacks, and then what kind of live food medley you can create to be optimal, or if there is a prepared food, what can be created there, what combination feeding regimen can help imitate it. But I don't know, just learning more about the actual mechanisms, as they say, of how this all goes down has been interesting. Okay, so yes, crucial factors influencing economic outcomes, with intestinal inflammation being a significant impediment to healthy development. Substantial evidence indicates that bacterial infections are a leading cause of intestinal inflammation in fish. I feel like this is the issue I've had with cribs a lot. Specifically, Aeronoma's hydrophila infection can induce intestinal mucosal damage, disrupt digestive and absorptive functions, and cause immune dysregulation, therefore negatively impacting growth performance, survival rates, and overall aquaculture profitability. Research shows that specific nutritional strategies can improve fish health, like dietary supplementation with alanine glutamine, which has been demonstrated to significantly enhance immune function in multiple fish species, including largemouth bass. Days becoming a few minutes longer for Mick. Let's go. While existing research has partially elucidated, I gotta look up this freaking word. Elucidate. Make something clear. Elucidated. Clear, explain. Okay. Research in the past has partially elucidated, partially explained the beneficial effects of alanine glutamine on fish health, its specific mechanisms in mitigating intestinal inflammation in Paperszzz daburanus. The loach remains unclear, the large-scale loach. As an economically significant aquaculture species, the intestinal health of this loach is linked to its growth and reproductive performance. All right, let's look at what they did. L-alanine, L-glutamine, and L-alanine were procured from Shanghai Uni Biotechnology Co. A basal diet meeting the nutritional standards for loach were formulated with alleglin supplemented at 4 graded levels, 0%, 0.25%, and 0.75%, and 1.25%. The complete formulation and nutritional composition are provided in Table 1. Perfect. The standardized procedure, all raw ingredients were ground, passed to an 80 mesh sheave, then blended sequentially in ascending order of their inclusion proportions to achieve uniform mixing. Interesting. Blended sequentially in ascending order. Following the addition of an appropriate amount of water and thorough homogenization, the mixture was processed into 2 mm diameter pellets using a laboratory scale pellet mill. They were then air dried under ambient conditions, packed in sealed bags, and stored at -20 degrees Celsius till further use. When I've been doing my experience of making my own foods, this is basically what I've done, except I don't have a pellet thing. I'm kind of just making strips. But yeah, I've basically been doing this. In theory, when you don't cook it, then it retains more nutrients. So that's like where the brine shrimp direct flakes is like, oh, it's low temperature cooked flakes. That's kind of the thought behind it. The mango rosy loach is my favorite. That's a good one, man. I love the rosy loach. I have some of these. They're very fun little fish. Good taste. So let's look at what they cooked up here. So we got fish meal 20%, soybean meal at 22%, cottonseed meal at 7%, soy protein concentrate at 16%, wheat flour at 24%, and soybean oil and fish oil at 5%, then 1% soy lecithin, 2% Ca parentheses H2PO4 to 2% choline chloride, sorry, 0.4 choline chloride, 1% premix 1, whatever that is, 1.25% alanine acid, and then each one has their own varying amounts of the allyl glutamine. This comes to about 40% crude protein on all of them, 7.5-ish% on crude lipid, and about 8% food ash. Okay. So they took 720 of the large-scale loaches with normal morphology and vigorous activity, sourced from the Loach Breeding Professional Cooperative in Fengshan City. Bro, what? That's an awesome name. It's coming up in food safety reports. I was hoping they had like a website, you know what I mean? But okay. We'll have to follow up on this later to see what they breed. But the Loach Breeding Professional Cooperative in Fengshan City. They are randomly allocated to 4 dietary treatments, supplemented with all the different groups each, and they replicated the experiment 3 times per group. So this is pretty thorough. Each replicate contained 60 fish reared in independent plastic barrels, 60 centimeters by 66 centimeters by 64 centimeters, to simulate the natural habitat and provide shelter. A moderate quantity of fresh water, hyacinth, Ecornia crassipes. Okay, that's just a common water hyacinth. Okay. It was introduced into each barrel. Prior to the formal experiment, all fish underwent a 7-day acclimation period under experimental conditions while being fed the diet that had none of the additives in it. So they were all fed the controlled diet for 7 days as they acclimated to the buckets. It seems like all the more well-funded experiments do have a period where the fish spend time in whatever, container they're going to be tested in before they actually start recording, start feeding them or injecting them with whatever's being tested. Okay, during the 70-day feeding trial, each group received their respective environmental diets corresponding to the supplementation level. They were maintained under natural light conditions and fed twice daily at the rate of 3 to 5% of their body weight. Water temperature was consistently maintained at 25 degrees Celsius, with aeration ensuring dissolved oxygen levels remained above 5 mg per liter. Weekly water exchanges were performed, replacing 50% of the volume in each barrel. Nice, 50% water changes each week. Regular cleaning was conducted for the barrels, freshwater hyacinth and feeding trays. Mortality and feeding behavior were recorded daily. So, here we go. Intestinal tissue samples were fixed in 4% paraformaldehyde, embedded in paraffin, and sectioned following the standard histological procedures. Oh man. So this is all the, if you want to do this yourself and cut them up, this is the section for you. All data was analyzed using SPSS. Oh, spreadsheets. I had to use this in a class once. IBM SPSS after verifying homogeneity of variance and normal distribution. One-way analysis of variance was conducted to evaluate intergroup differences. Okay. Results. Let's see. In the control group, where nothing was added, normal intestinal morphology with intact villi and well-organized muscle layers was observed at 0 hours. So this is when they'd just been injected. Pathological changes emerged at 6 hours post-infection, manifesting as villus structural breakdown, swollen goblet cells, muscle layer loosening, epithelial necrosis, and capillary congestion. Jesus Christ, it's like I just took an antidepressant. The most severe damage occurred at 12 hours, characterized by extensive villus loss and detachment, a disrupted striated border, increased goblet and columnar cells, muscle necrosis, and heavy inflammation infiltration. By 24 hours, the intestinal structure showed clear signs of recovery with substantial improved integrity and reduced pathological features. Okay, so the control, some of them seem to recover some, and we can see the stain here of it pre-infection to post-infection. I don't have any way of understanding what I'm seeing, but I do see that there does seem to be a difference between these three, these four. I can't even count. Okay, in the 0.25%, the 0.25% allergen group, the intestinal tissue of the loach exhibited a relatively thick muscle layer and intact villi prior to the infection, or what they're calling the challenge. At 6 hours, while the villi remained largely intact, the muscle layer displayed significant loosening, disorganization, and fragmentation of muscle fibers. By 12-hour, severe villus damage was evident, characterized by extensive shedding of columnar and goblet cells. Barbara says, sounds like my last GI checkup. Jesus. Marked inflammation infiltration. Inflammation infiltration is killing me, dog. Like, yo, my foot is inflamed. I got that inflammation infiltration. By 24 hours, the intestinal structure had substantially recovered, showing restored integrity with neatly arranged villi and epithelial cells, indicating a return to histological normality. All right. In the next group, the 0.75% allergen, again, at 6 hours post-infection, they demonstrated superior architectural preservation compared to the previous groups, showing minimal villus damage and maintaining a compact, well-organized muscle layer. By 12 hours, moderate pathological changes were observed, including partial detachment of columnar cells from lamina propria, irregular serration of the striated border, ruptured goblet cells, loosening of muscular layers, fragmentation of muscle fibers, and slight intramuscular hemorrhage. Well, just a slight intramuscular hemorrhage. 24 hours post-infection, intestinal morphology had recovered to a state of structural integrity. Okay. So from 2.25% to 0.75%, there was a significant difference. So every little digit counts, man. And here we have again the 4 photos at hour 0 of infection, 6 hours, 12 hours, and 24 hours of infection. These are pathological intestinal sections. Magnification X100, X400. So here we go, the biggest percentage group. Big Steve said, when can we go back to talking shit on everyone and making people mad? This shit is for nerds. Who do you want me to talk shit about, Big Steve? I need a target. I got no one on my mind right now. In 1.2% allergen group, paramysgyrnis, the loach, displayed largely preserved intestinal architecture at hour 0. At hour 6, the restoration of the villus striated border, presence of erythrites among columnar cells, damage to goblet cells, and minor hemorrhage. By 12, severe structural disruption. Lost striated reporter. Oh my god. 24 intestinal morphology shows substantial recovery. Okay, okay, okay. Tell me what to think. Steve's getting antsy. Steve is getting absolutely antsy. Okay. Hold up. Hold up. Let me get to the conclusion here. Okay, Steve, this is what you want? This one paragraph? You want me to skip everything and read the conclusion? Fine. Let's see how nerdy this is for you. Dietary supplementation was 0.75% and 1.25% allergen significantly enhanced the antioxidant capacity and nonspecific immune performance of the large-scale loach, while also preserving intestinal tissue integrity. Following Aeromonas hydrophila infection, allergen facilitated the rapid activation of specific immunity through multiple pattern recognition receptor pathways, thereby promoting the clearance of pathogenic bacteria. Additionally, allergen may enhance the tissue repair function of IL-6, though this mechanism warrants further investigation. Barbara asked, what caused the changes they talk about? I missed that part. The changes like the rupturing and all that stuff, that's the infection. Yeah, the Aeromonas hydrophila. A heterophic rod-shaped bacteria mainly found in areas with a warm climate. It can survive in fresh or brackish water. It can survive in aerobic or anaerobic environments and can digest materials such as gelatin and hemoglobin. It was isolated from humans and animals in the 1950s. It's best known of the species of Aeromonas. It's resistant to most common antibiotics in cold temperatures and is oxidase and indole positive. Also has a symbiotic relationship as gut flora inside of certain leeches. They're gram-negative straight rods with rounded ends. They can grow at temperatures as low as 4 degrees Celsius. It's toxic to many organisms. When it enters the body of its victim, it travels through the bloodstream to the first available organ. It produces aerosin, a cytotoxic enterotoxin that can cause tissue damage. It's considered to be an opportunistic pathogen, meaning they rarely infect healthy individuals. It's widely considered a major fish and amphibian pathogen, as pathogenicity in humans has been recognized for decades. Its genomic insights could be a stepping blah, 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 blah. Okay, yeah, that was the changes. So a little bit of a dietary supplementation and enhance the outcomes. Let's see. Let's see if we can look at a chart that Steve can approve of. I want to see a chart that shows us how much it made a difference, because all this gene expression stuff, like some of these charts I'm not going to understand, you know. So I need the chart that shows me like what percentage good and what percentage bad, you know. That's what we can do. So want to go swimming in that local pond? Absolutely not. Absolutely not. Loaches that I'm interested in. Let me get to that. Loaches I'm interested in. Some hill stream loaches. Um, there's some loaches that I think I think might be coming into some stores sometime soon. Let's look at those. Let's look at some of those. Barbara, you're gonna go. Nick's gonna go swimming in a local pond in a mankini. Mankini. Okay. Feast your eyes on Noella latermaculata. Okay. This was recently in Practical Fish Keeping magazine. Okay. When was this published? Well, I said it was recent, but I can't say when it was published or not, so I guess not. I guess not. I'm trying to find a good photo of them. We got some pectoral fin photos. Yeah, this is not one that's as I think its name is the Chinese snake loach. It's the common name. So let me try that. Chinese snake loach. No, we're not talking about the freaking fluey loach. Common names, once again, useless. Well, here's a good photo. What kind of website is this, bro? Fishbiosystem.ru? Well, anyways, Chinese snake loach. This is pretty cool. Nick, we just decided you a new task. Yeah, that's not gonna happen. This is, you know what? I'm kind of sick of the snake looking loaches. I mean, this loach looks cool, but I've been thinking about Befordia quishuensis a lot more recently. Okay. I've been thinking about this one a lot more recently. It's the body shape, it's the featheriness of it. Okay. It's like Soelia, but better. You know, a little bit bigger. I want some bigger loaches, man. I like big loaches. And I lie all the time, but this is the truth that I'm saying. Okay, another thing is, so Pseudogastromyzon laticeps, right? This is the purple fin one. Barbara said, I don't want Nick to have intestinal lining that looks like marbled pastrami. I appreciate that. So I know this one as the purple fin. Like the Pseudogastron laticeps, like, okay, this is the one with the purple fins. But like apparently there's ones that got blue fins. That was kind of news to me. I'm kind of into the blue. I'm not gonna lie. I kind of like the blue fin. It seems like it's got a bit more of that green in there. Like there's something about it. I might kind of like that more. Next says, but why not? It'll guarantee you many views. Trust me, bro. I'm not trying to guarantee many views. That's not my motivation. Man, we'd have to do a lot more clout chasing. Yo, what the hell is this? This is a mislabeled photo. Pseudogastromyzon SP Redfin. Yeah, this is really useful. Thanks, Glasser. Thank you, Glasser. Are they always blue fins or is it only true breeding colors? I don't know. I haven't kept them. I'll be honest. I've seen this fish in the stores a few times and I've never left with them because they look terrible in the stores. Now I understand stores not the best place, but I think they do got to grow a bit. I heard Father Fish said you're a punk. Are you gonna take that? I mean, are you joking? Or like he actually said I'm a punk? I mean, what am I gonna say to that? I'm a punk? Well, he's a con artist. Which which one do you want me to take? Which one? I'll take punk. I guess I'll take punk. You know what, Father Fish? Let's see who's alive in 10 years. Okay. In 10 years, we'll meet back up. And if you're still there, I'll accept this title. Otherwise, and I'll say I'm sorry for calling you a con artist. Until that happens, you're a con artist, S tier scammer, and maybe Steve is just making up that you called me a punk because he's getting bored. But there you go. I'm reiterating what I've already said. Nick's a fish room punk. It's an honor. I'm not a fish room punk. Moonlight Aquatics. Moonlight Aquatics. New local Austin said exactly what exactly what a punk would say. Moonlight Aquatics. Why are you saying Moonlight Aquatics, Johnny? Is there something specific that you are looking at? Oh, you know what? I do think I'll be able to find the loach that I want on here, though. Was it Louise that had him or something? I don't know who it was that had him recently. I'm gonna just sort by price. Can I sort by high to low? Because I'm sure it's expensive. High to low. Steve did $2 donation, said epic rant. That's really, Steve. That's what you want from me. That's what you want. You just want me to insult people? Jesus Christ, man. Did you just make up that I was called a punk? That's honestly a good strategy if that's all if that's the entertainment that you desire. I mean, it's not a bad strategy. Luckily, I just, you know, you said you made it up. Yeah. Well, I mean, I've said all those things before, so I'm just impressed you said punk because like that's like such an old person thing to say. Like I could almost believe it. So this is one that I'm really a fan of. Okay. The Gastromyzon leptogaster. This is what I want. This is what I want. I like this one. This is on the list of expensive hill streams that I would consider buying. This is one of them. Now, I'm not ready to buy this one yet. I gotta see how much money I have left after this swap. This swap is gonna absolutely demolish my fish budget. Yo, Steve, did you see the raffle items we just announced for the swap, by the way? I don't know if you were here in the beginning. Just since I was reminded, but yeah, Tropical Fish Co. donated three $100 gift cards to our raffle. So it's got nothing to do with the fact that I'm on this website. I was looking for loaches, but it did remind me. It did remind me. Shout out to them, man. That's pretty freaking cool. So we're getting a pretty loaded lineup. Now, one thing about Loaches from Tropical Fish Co. is nice is they're going to go through the extra effort to identify them properly. I do kind of like these golden stripe hill streams. I do. But yeah, Gastromyzon, Liptogaster. Barbara says, yeah, dude. I'm gonna have to make the biggest tropical fish co order I'm capable of. I don't even know. But the board was super excited about it. So yeah, the pressure's on for this swap to go well and be run. Like I kind of have no excuse for it to go poorly. We got a board of people that are really invested. We seemingly have a venue that is usable. We've got a large assortment of raffle items. So in theory, this should be a successful event for vendors. It should be a successful event for companies donating stuff. It should be a successful event for us raising some money off of memberships and some raffle tickets. And yeah, so I'm like, it's getting closer and closer, and I'm just hoping it all goes well. Mick said, "Don't let anyone say we didn't have a great time tonight." Yo, Vic is so triggered by your comment about the nerds, Steve. You gotta let me know that other people are appreciating this that aren't you. Yo, who can I insult? Hold up. Yo, we got it. Steve donated $2. Like, what's $2 worth of fish content? Zebra said, I can't wait. Dude, I'm super excited. I'm gonna go over the vendors and some of the stuff they can be bringing us. I could do that now, or I could look for someone to start a beef with. JP Jeffrey says, Nick, freshwater ichthyology would be a great guest for a catfish deep dive. Seems like you're kind of guest. Yeah, I'm a big fan of freshwater ichthyology. Rebecca, like I said, I saw her talk at the Clash. That's a good question. I don't know. See, the thing with Featherfin is, like, I had talked to him. I usually do streams with people that I've already like interacted with and talked to with. So it's not very often that I do a stream with someone that like I've just never talked to at all. Um, I'm trying to think back to everyone. Like I think everyone I had talked to some extent. I just haven't talked to Rebecca before, so that's the only reason I haven't done it. Mick said I'm not bothered, the 1%. Okay, aquarium fish. Let's see what pops up. Oh God, not the aquarium store depot. This is added to my list of content slop channels. Content slop. Oh man. but he's dominating the search right now. You put aquarium fish, it's all this content slot factory. We get keeping fish simple. At least one in there. Paul Cafaro. Oh, this is one of the Florida boys right here. Jesus Christ, that was loud. I'm so sorry for that. Rebecca Wright, she's awesome. Yeah. Okay. Let me look at this list of fish real quick. So I got Jesse sent me a list of fish that he may be able to bring. Okay. It's quite a long list. I'm just gonna let you know. But I do think there are some interesting things that we might want to look at that is of general interest. Okay. So here's a list of fish that GSC had just one more fish said that he could bring to the swap. And I'm gonna be honest, like this, I haven't been to a lot of swaps. I really haven't. But I'll be honest, the list of fish that I have access to purchasing coming up at this swap is frightening. I don't even know what I can only buy so many fish. Like I got to be responsible. Um, but there's a lot of bangers coming up, so it's just like, bro, I don't even know what to do. So he's got listed here African red-eye tetras, Congo tetras, diamond tetras, glow light tetras, red cap moon tetras. These are they're kind of a rare or niche one that you don't see around as much. A little bit too minimal for my taste, my personal taste. Like I appreciate them. Barbara said tetras don't usually show up at swaps. Cool. Okay, Barbara, you know the swap scene, so you'll be able to evaluate what we got coming up here. Okay, but yeah, it's a little bit more minimal than my taste, but like I can appreciate. Yeah, this is a cool rare the red cap on top. Like you could see how they can make a nice little display with those. The Buenos Aires tetras, red phantom tetras. These I think we talked about those on the other day. Those are cool, but the jelly bean tetras are one that I definitely really believe in. I think this is a solid fish. Like if I was gonna list some small tetras to breed, when I saw them in person, they look fantastic. The videos that Aaron sends me of his look fantastic. Like as a small Polaris fish with some bright neon coloring. I mean, what else could you possibly want in a tiny nano fish? You know, and it's African fish, and I have a slight bias because there's not as many tiny little African fish that I get my hands on. So that's definitely a cool one for me. Catfish, we have tiger whiptails. What's a tiger whiptail? Tiger whiptail fish. Okay. Ooh, it's like one of these things. Okay, pseudo hemodon sp tigre gets how big? 5.5 inches. Holy. The tiger face loricaria is a smaller growing member of its genus. This is a small one, okay. From the Amazon, still undescribed, has a unique pattern, sandy stretches, moderate to high movement of water, open areas of substrate. Barbara said the colorful ones will go well. The duller but more rare ones will appear to one person or 2 to 3. If the latter, you may get a bit more. If not, yeah. This is all just swapping and swapping. So no auction. Although now that we have our bank account set up, we're incorporated and all that, we can now do an auction properly. So I do think we'll try and do a big spring, summer, something. I don't know. We'll try and do a seasonal auction, and I'm pretty excited about the prospect of that. But let's see if we can pull off this swap. I do feel quite a lot of pressure. Okay, we got Geo Red Hump. Geo Red Hump. Oh, Jesus Christ. Okay, this is the one that Bob would like. Geophagus Steindekern Canary. It's got one of those faces only a mother could love. Now shut up. Show me the fish. Shut up. Show me the fish. It's fun when two people want the same not popular fish. Yeah, it is fun. Um, okay, Mick said egg carrying catfish. Which one is that? Oh, the tiger whiptails? Yeah, I think they're the ones that like they have their eggs in the sand with them, kind of a thing. I believe. Is that what you saw? Egg carrying? Yeah, yeah, yeah. I've seen some videos, someone breeding them. Tiger whiptail fish. But yeah. Okay. Full breeding behavior. We have the Sino flavitiniatus. What the? This looks like a quarry. How big does this thing get? Rarely grows larger than 5 inches. Okay, that's still a little big. Hognose brocus. I'm out. I'm out. Yes, I've seen this one. I don't like the brocus. Albino hoplo catfish. Okay, I'm listening. I'm listening. Male takes eggs in mouth. Beard carries eggs still hatching. Hoplo catfish. Black marble. Okay, okay. Okay. And Otto. Okay, we got the pink glow angels. I saw that in this video. Ooh, albino threadfin acara. This is the one. Were they saying whether it was this versus the geophagus, the albino geophagus? Which one looked better? This does look pretty cool. Now this is just unachievable. Like I'd never be able to. Hoplos are nest building catfish. Mick said. Okay, nerd. I'm gonna be like Steve. Nerd. Those cars are pretty. Yeah, the threadfin cars are pretty. Now I cheated. I did look this one up before the stream. These J barbs, yo, these J barbs though. I, I'm just gonna tell you guys, I'm going down a barb hole. Okay, the blue banded barbs, they're kind of cool, but man, they're so mid compared. Now I'm looking at all these other barbs. I got to school like 20-ish of the other barbs, maybe 20 to 30, because I started with like 10, and then I bred a bunch. I took some to the auctions and stuff, and I just put the rest in there. I'm just keeping them. I don't know if I'll try and breed them a bunch. Um, I only have like one adult male, which I think was, anyways. Jesse has J barbs on his list. He said he has them. What's up, Jose? Man, this fish looks dope, and it's another. I think it's an African barb. Yeah, Equatorial uh, Guinea, Democratic Republic of Congo, Cameroon, Gabon, indigenous to West African countries. So like bright, West African. That's me. Now this is nice. This is nice. Very diet. Okay, okay, but no, this is a nice fish. So he better bring some bags of this. Now I don't know how often it's that fired up or not. You know what I mean? But man, um, my LFS showed me is what is that? J Tibet. Did you spell something right? Is it these? Well, if it's these, I like what you said. You said much more red when colored up, just like Barreras can be. Huh. I just got barbs for the first time this year. Started simple with cherry and rosy barbs. So far, I'm loving them. Yeah, yeah. Like these barbs, the butterfly barbs are two I'm pretty interested in. I was kind of interested in the melon barbs, but they're a little bit bigger, and I kind of like these smaller ones. Okay, we got some Berkshires. He's got the award-winning Ocean Blues, Vampire Shrimp. I might need some of those, and freshwater river prawns. I wish I had something to put these in because they look nuts. And people actually farm these for food. So maybe you could start like an aquaculture side hustle with these. But I mean, this is this big ass shrimp with ridiculous cartoonish uh claws. and definitely would stand out in the aquarium. They get pretty big, so kind of like a showpiece situation, like a larger fairy fish, but pretty cool, pretty sick looking. Congo spotted puffers, the dragonfish, African butterfly fish. I've always been curious about those. Clown killies. I need to try clown killies again sometime soon. And fish room punks, man. Fish room punks. And he's got all the envy foods. That's pretty dope. The other person coming is, God damn it, bro. I can't spell. Sabu. I know how to spell. I've been to his website a billion times. Saburai Aquatics. Yes, 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 yes. Saburai is coming. I'm not sure exactly. I don't know if he posted everything that he's bringing. Now I need my filters, Sabu. Where's my filters for in stock? But he's all got all sorts of fancy plecos and catfish. He's all has some really nice snow white pandas. I saw Queen Aberesque and tiger plecos. So that's kind of the vibe that I've gotten. He seems to be the fancy pleco individual. Quite a lot of fancy plecos. And some other quarries. See the orange Venezuelans. Shall you ask Jose about the taurine, Nick? Oh, go ahead and ask, Nick. Come on. And the heat shock proteins. Yeah, we could have used you earlier. We were trying to read some papers. They're injecting loaches with taurine to see if it helps with ammonia exposure. Holy. Oh man, I'm like, this never happened to me. I'm like, my voice, I get tired. I ain't drink enough water. I'm not drinking enough water. I'm trying to find my freaking list. Not everyone has the accessible website. That's a problem. Louise is coming. I know she's doing pre-orders, and I know that there's some fish that she got that I'm pretty into. Let's go to the categories here, because let's see, catfish. Let's do barbs. Let's do cichlids. Let's do cories. Let's do all the fish basically. Why not? Well, let's just do everything. Let's just do it. I know she's got some killifish I want her to bring. That's for sure. I know she's got some killies. She's got the Australibris Nicoprinus. Freaking availability in stock, please. With the catfish. Do I want any of the? No, no. Let's go straight to the cories. Hold up. The Corydoras. Nick said I'll ask my doctor. I thought you said I asked my D friend about it. What the hell? Okay, gold lasers, Histatus, albino sturbi, Hesperosis, sturbi, Brazilian spotted ottos, redfin otto, Similis, skunk cories, Peruvian striped ottos, Palladius, red Venezuelan hyphen, galaxy ottos. Ooh, yo, Steve, should she bring more galaxy ottos? Axolotl corridors, the polka dot, the haplosoma, Atroposonatus. I think that this is the one that Aaron was asking her to bring. The slate corridors, the blue concolors, so not the new ones, but a different slate concolor ones. Gold spot, dwarf pleco slash autosynclus. Oh, para autosynclus caroensis. She has some of these in stock. I need some more. I got two. I need more. Okay, reticulated corridors. Well, they have some cool stuff. I'm looking at the website now, looking to yeah, yo, get your pre-orders in on all these places because I know that Jesse said he was bringing like 6 bags of whatever he brought. So if we do have good attendance, I could see some things getting snatched. Uh, but no, Louise got all sorts of cool stuff. So I don't know what she's gonna bring just on her own accord, but I would recommend if there's something you want, get your pre-order in for sure. And the same with Jesse too. And Sabu, all of them. Hit these people up. I gotta get my links together for some of the other. Oh wait, I didn't do by availability. Hold up. Hold up, hold up, hold up, yeah, yeah. Tropical loach, weather loach, dojo loach, Abditas Borneo Hillstream loach. That thing looks funky. Dwarf chain loach, golden zebra loach, red squirrel loach. She's got the purple fin hillstreams, the laticeps, brown mosaic hillstream, red zipper, rainbow tiger, butterfly Borneo loach. Flashes fish tanks to what fish you getting, Nick? I don't know. I don't know if I'll ask. I might ask people to bring things. I don't know if I'll pre-order it. I might like ask them to bring things, and then if no one else buys it, I'll buy it. Like just give other people a chance. But we got Tweety's Hillstream Loach, and we got Wooming Jade Tiger Hillstream Loach. What the hell is this, Louise? Where's the photo, Louise? God damn it. Specific. Oh, she got to bring these. Virfordia viridis. Is that one of the ones? I'm gonna have to see if some other people are getting these or not. But holy, yeah, I might be into that. I'm discovering I like the Bufordia. I like, what is it? I like the shake of your jib. The hell is that saying? Someone tell me what the saying is. This kind of looks like the one that I want though. The Leptogaster. Like it's got the green. Wait, I just Googled this and Louise's photo is one of the top results. The hell? It's crazy. Instagram post, one of the top results. That's some good SEO, Louise. Good work. Good work. I'd like to get some. I wanna up my loach game soon. This is one fish I'll ask for pre-order. I think she should bring all the pairs of that she has because I think she'd sell them all. The Australorhynchus nicoprinus. I need another pair of those at least. She has two different Procatopus that are both really nice. I don't know how many people would be interested in those. But it's possible. It's definitely possible. The chocolate Australs are kind of cool. Albino Gardneri are kind of cool. I don't know what the neon green lampi killifish are. Neon green lampi. A macrophthalmus. Okay. Looks a little bit different. Okay, I like pencilfish. Peppermint gourami is on your prior list as well. Yeah, I don't think anyone has peppermint gourami that is coming, but goddamn, I want that fucking fish, man. That's a cool fish. I like these diptails, all the purple fish. Yeah, these are all nice. The Cinepa Super Red Type 3s, these are all cool. Ooh, three-line pencilfish, they're trifasciata, I like those. I like pencils. Even if I like not even just I'm not really trying to breed my any of my pencils right now. I just enjoy them. They're good dither fish and they just look cool hanging out at the top. Platinum half beaks, black tiger batis, pandagar. Pandagar are cool. People probably would buy pandagar. I should buy them. Oh, purple spot gudgeon. That's a cool one. Ooh, the toothpick fish. That would be a cool niche fish to bring. You probably don't see them so all the swaps. Indian dwarf mud skipper. She probably could sell those. I don't know how many people would be ready already for them, but I know that people do want the Indian dwarf mud skipper. So I bet if they saw this, they probably would pre-order. Ooh, Siamese glassfish. I do really like these actually. These are cool. Yeah, I probably need a dedicated tank for those. These Siamese glassfish I like quite a bit. No, no, not the injected ones. No. Baddest, the Blenny. Okay. Oh, okay. I did see this, and I have to wonder if I'm even prepared to take care of these. But she has it listed that she's got Blue Amur Taiwan bitterleads in stock. I mean, this is a photo from inside her store, and they don't look like they're fucking dying. So I think she should bring these. These would probably go. I mean, if no one bought them, I would buy them. I might actually just buy them off the gate, um, because I often do the bitterleads come along. And I can't be choosy. And specific bitterleans I'd like, but what the fuck? I can barely get any. So this one does look all right though. Um, Blue Retiring Bitterlean Aquatic Arts has them in stock, and these photos don't look too bad. Now the thing about bitterlings is supposedly they're like an algae eater. Maybe they eat some parasites and stuff too. I don't know. So that might be a potential reason why they're kind of cool. Parrot said, just popped into. I think I should pre-order a tiny core species or see what all the vendors bring. You could see what all the. I mean, it's up to you, man. If you know you're gonna buy them, then there's no reason not to get them. But if you're thinking, well, if I go there and see something I didn't know I wanted over them, then I would maybe wait. But um, someone's got to bring some status, right? Right? If you think you're gonna buy them, add something else. I already explained my reasoning. I don't know if I'm going. The only things that I would pre-order is stuff that I don't know if they're gonna for sure bring or that I want to make sure I get. Besides that, I kind of want to go in open. You know what I mean? But there's definitely a few things that if I saw pop up, I'd be like, holy, I want that. I want that. I need it. What's up, George RB3? How's it going, man? How's the Christmas cards been going? George has very cozy streams opening Christmas cards. It's kind of wild how many he gets, but it's pretty dope. Someone needs to send him a real $100 bill though. Someone sent him a fake $100 bill. I was like, why would you do that? We gotta send him a real one. Oh, these are two cool Tetras. So the Black Darter Wismani, these are supposed to like behave like cichlids, kind of like cave spawner type situation. I think they're dope. This is an example of one that I would ask. It seems like many stores are waiting to ship during Xmas. Best to pre-order all fish. Well, or all fish could be sold Jan 4th or so. Maybe. Yeah. This is one that I'd ask for her to bring, probably, if she's got a decent amount. So, Louise, if you see this, bring those damn weird ass darters. The Wismini Tetramorphos. One of those. The hummingbird tetras, I think, is an underrated one. This is on my list of things to breed eventually. They're kind of clear, but they do actually get a decent amount of color later on. Like you can see some of these photos. Like I've seen them in person. Like they actually do get a decent amount of color, and the males get a pretty extended dorsal, which is kind of nice. Shit. Got some red cherry, red line, all these touch. God, Louise got some fucking fish, bro. It's ridiculous. Oh, that's a good picture of the jelly beans, so you can see the color in there. Not bad, not bad. Yeah, excuse me. Pink lemonade tetra, 36 a tetra. Jesus. Oh, I'm too broke for this. George said, yes, send me a real $100 bill. I'm working on it, George. I'm working on it. Orinoco anchovy tetras. Microsymobrycon CF cassiquare. What the what is this? What is it? Toothpick fish is possible to breed if pain and some effort. Okay. We've got some random niche clear fish. I don't know what this thing is. I'm looking for a good photo. Okay, well, that's like a deceased one. Okay, here we go. This is a bit better. Interesting. Coloration in life. Cool. I like clear fish. They're fascinating. They're fascinating. Absolutely fascinating. Send George to the Clash. That'll work as well. Oh, send George to the Clash. Yeah, yeah. Forget Johnny, man. We don't need Johnny at the Clash. I mean, we need Johnny at the Clash, but Johnny, like, get your bread up, okay? George, let's send George to the Clash. Holy. Oh, goodness. Ooh, checkerboard cichlid filamentosa. Hold up. Hold up. It's kind of cool. Rainbow Severum. Oh, Vanacarb immaculata. That's a cool one. Laticar aguirre. Geophagus wine milleri. I have anchovetas in a can with tomatoes. Barb says, isn't Johnny much closer to the Clash these days? I don't think he's in Iowa yet, but I do think that the drive was. Ooh, she should bring these, the Tenacara Candida. I need more of these, actually. I need more of these. God damn it. I need too many fish. I need too many fish. This is getting ridiculous. Oh, she's got the gold convicts. George said I used to have checkerboard cichlids. They're fun. Got the giant checkerboard cichlid, a juvenile blue acar, just the normal blue acars, not the electric blue. Okay, I know Snyder's Aquatics is coming. Do they have like a? See, some of these places I can't find like a website for. Like they just like they only do the swaps. You know what I mean? So it's hard for me to tell. So let's look at a past stock list. A past stock list was some guppies. Okay, so nice. We don't have a fancy guppy person there. Some plecos, some shrimp. Okay, snails, and some plants. Okay, what other stock lists have they got? Let's just see. Okay. Guppies. Okay, they got some endlers here. They got some diamond tetras. So they got the same plecos, shrimp, snails, and some plants. Okay, fantastic. Okay, we got zebra aquatics. And does Andrew, does he have, he has a Facebook page. He got a bunch of cool handmade caves and cool plants. Ooh, I need some Laganandra or something. That's what I need. No, what are the other plants that zebra aquatics got that I need? Oh man. Do I got a list? Temporary product list. Okay, Choya wood, plecocanis. Okay. Man, is this better not? Yeah, this is from last December. Come on, Andrew. Come on. Yeah, where's the update? It's been a year, man. It's been a year. Where's it at? Oh, yeah. He's got variegated boost and Anubias. Oh, yeah. The large checkerboard change gender if needed. Lots of research on those. Yeah, I thought I saw something that some of the other ones might as well, which just makes everything so much more interesting. I'm a male. Nope. Okay. You gotta suit the needs of the group. Okay, Carol's beta, betas. Carol's betas, like beta betas. Okay. I have a functioning brain. I can do this. Carol's betas. Now, let me see. I'm gonna assume this is a Facebook page situation. Carol's betas. Come on. Come on. The digital footprint is not footprinting. Okay. She has a wide variety of beautiful imported betas, including Hellboys, Giants, multicolor, and more. This is the old post. Okay, so this is one of those exclusive vendors, that you just hear whispers of, but the stock list evades. Carol's Betas mix says, yep. Okay. And then, what ones have we not? Is there ones that I think the other two that we might potentially announce yet does not have websites either. So I can't, I'm out of luck, man. We already went over Jesse's list. So I think we covered everything. I think we've covered everything. Mick said the Bettis thing was smart beta name. Yeah, I'm excited to see what kind of Bettis. Some of the alien Bettis do look kind of cool. Now, shout out to Jesse. Um, link in the chat while I'm at it. He's been listing more stuff. Now, interestingly, I didn't know that he was getting into decor sales. This is next level stuff. Now we got the merch coming in. Okay, we got the towel, we got the hat. He's listed these catfish. Okay, he listed the tetra. All right. The ninja wood cats. We got plants. A couple fish said, "Okay, I'm back. Anything big I missed?" Well, where did you leave off? Uh, what was the biggest thing that you could have missed? I feel like I went through the paper on the secondary sexual dimorphism of the Botidae family. I thought that was really interesting. And potentially could help sex out some other loaches or just give ideas of what to different things to look at for sexual dimorphism, I think. I don't know if you were there for that one. Yeah, first evidence of secondary sexual dimorphism in freshwater fish Botidae. As far as loach cool stuff, I thought this one was kind of interesting. The one on the loaches in the Mediterranean was cool too, on how they change microhabitats with the different flow levels. What's up, Sleeper BBQ? What is up? Holy man, yeah, I'm super excited. I'm super excited. That's really all I have to say. Zebra says, I have big plans for 2026, a website and a bunch of new products. I'm looking forward to it, my friend. I'm looking forward to that show. I know we're gonna have we're gonna have a terrestrial isopod vendor, and that's a pretty cool list. That's a pretty cool list of stuff. The mankini part, Nick promised to dive and search for shock proteins in a pond. I definitely do not. I definitely do not agree to dive for heat shock proteins in any body of water. I'm actually not willing to look for them at all. I don't think I should be the one that is relied on for this. Okay, we got some terrestrial isopods coming through. I don't think we've announced this vendor yet. It's just an individual that I happened to come across. I think she was a friend of a friend and got in a car accident, so is trying to raise money for a car. Just happens to have a bunch of isopods. I don't know that she's ever really done swaps that much before. But here's a list of some of the isopods that she might be able to bring. And this list includes some Porcelio that I've been looking for and some morphs of them that I was not familiar with. So Porcelio hoffmannseggi is one of the bigger isopods. I like big isopods. These are like the titans, okay? They call them the titan isopod. And I like it because I just think that it being Porcelio, you always hear like Cubaris and Armadillium. So I'm like, yo, some fancy Porcelios. And they get a little bit larger. They're from Spanish. They're Spanish, I think. Parrot says she does the Baltimore plant swaps a lot. Okay. She does the Baltimore plant swaps. There we go. So Baltimore plant swaps, but who knew that the aquarium swaps might want some isopods too? Like Steve was like, let's see that list, you know? So yeah, kind of a little bit of a fancy big isopod. I'm trying to see what the freaking size is. The Baltimore plant swap does sound like a size. Okay, so they can get 1.5 to 2 inches long, which is pretty big. Now, what I didn't know about was the morphs of this isopod. So she got the I want Warneri. I know Warneri is the one that I spent a lot on recently. I I think she has Warneri, but I don't know if she's bringing them because it's not on this list. But I should ask her about Warneri again specifically because I saw a different list of stuff that she keeps that was on there. Um, but Warneri is cool. I have the Dalmatian Warneri. Porcelio Hoffmannseggi orange. So like one thing that's cool about these, besides the fact that it's a pretty color, is just the size, right? So a lot of isopods, you see the pictures of them. Like I feel like the zebra isopods are ones for me where like they're really small, man. The Armadillium maculatum, like they're pretty small. They're cute, but they're small. I like things that I can see a little bit easier sometimes, so a up to 2 inch isopod is appealing to me. But yeah, these are a little bit fancier. You see a quantity of 5, they're selling them for $65. Okay, talking about bugs. You guys don't know about this kind of luxury. Okay, now the Pak Shongs I like a good bit. So she's got the Cuberus red Pak Shong. These are cute. Everyone likes the Cuberus because they got the little They got the little faces. I mean, look, like this is very. Look how angry this thing looks. Why it looks so mad, bro? Cuberus platane tongue sung. Oh, this is a fancy one. What? Yeah, I know that's not $95 for one isopod. No, get the fuck out of here. No way. Okay, yeah, this is more reasonable. It's got some cool orange on it. Okay, Cuberus orange freezy. Cuberus orange freezy. Ooh, ooh, ooh. And it's reasonably priced. My pineapple spikies are multiplying. Ooh, very nice, Steve. Very nice. The spikies they're cool. I I think they might be a little small. How big do they get? Ooh, this is a good orange. This is a good orange. I might pick this one up depending on how active it is, because this color is phenomenal. Yeah, yeah, that's a good one. Okay, Cuberus SP Miyako. They're small. Okay, so Cuberus SP Miyako. Okay, little cool, little chocolatey coloring. It's no orange freezy though. That's the top Cuberus for me right now. The red ones are all right. Cuberus Happy Nun. Nope, nope. It's cute, but I'm searching for the next. I'm searching for the next orange freezy. Okay, now we're talking. Cuberus Cappuccino. This is kind of like a Panda King vibe. Pretty much all the Cuberus are small. Yeah, they're small, but they got the faces. You know what I mean? Yeah, I can get with this. I like this. Is there a Mad Bull isopod? I have no idea. Probably. They make up all sorts of names. I'm not familiar with this. Carabodillo martinicensis. Martinique. Ooh, just described in November 2025. New species to science. What? Yo, we're on the fringes of science. Yo, what the heck? It's got like a roach shell to it. Nah, this thing's looking nuts. What? What? Oh man, and they roll up. They roll up into a ball. That's what people love that. I want Cubaris White Shark too. Oh, Steve's got his list. I don't know, man. This one might make my list. Look at this thing. That is crazy. Oh, we got the most accurate description. I'm putting that link in the chat. That's nice. Okay, Armadillium versicolor Ukraine locale. Okay. Okay, interesting. It's like a blackish thingy. More rubber duckies. I wonder if Chad might bring some more rubber duckies. Okay, bring in the zebra, of course. Oh, Armadillium gesteri. Can't ever go wrong with the gesteri, man. This is definitely one of the most appealing isopods. I have some of these. They're big, they're colorful. You see them walking around regularly. Don't get much better than that. Crane pied woodlouse. Well, I kind of like that coloration. Kind of like that. Okay, yeah. Porcellio prunosis or Oreo crumble. I'm obsessed with my Gesteri, Parrot said. Yeah, I don't blame you. Okay. We got the orange powder, the blue powder. I was kind of looking up ones that I don't know. Cinadillo Vietnam Dysphoria. She gave me a lot of them. Nice. Okay. It better not be this roach. Where is this freaking? Can't even find a good picture of what this is supposed to be. Yeah, I'm not looking at roach photos. Okay. I'm just gonna have to see this one. Trichorhina tomentosa dwarf white. I like the sound of that. The dwarf whites. This is one I was thinking about maybe using as a feeder because they're smaller. Okay, Troglodillo SP Sunset. Oh, hey my. Fast reproductive species. I like that. I like that. Yeah, the face just doesn't get old. It doesn't get old. Shit. All right, all right, all right, all right. So now the swap is looking pretty fantastic. We're gonna have it all. We're gonna have it all. Okay, guys, ladies, individuals, things that exist, aliens, and also criminals, I assume. It's time for me to sleep. Well, it's time for me to get off, take a bath, eat some ice cream, and then go to sleep. Ooh, ice cream, peanut butter, vanilla ice cream, peanut butter, additional milk so I can make my ice cream soup. Yes, this is what I must do. This has been a very educational stream for someone. I didn't learn a thing. I didn't learn a damn thing. No, seriously, this has been a good stream. Look, if you're in the area, god damn it, man, I need emotional support. All right, I want this. I want this swap to go well. Luckily, it seems like everything's lined up for it to go decently, or at the very least, there's going to be some pretty dope people, which is what it's all about. The raffles, the fish to buy, that is all excellent. But I am mostly excited about the collection of people shaping up to be in the room because honestly, we all do not get to hang out with people as nerdy as ourselves when it comes to fish often. And there's going to be a bunch of pretty fish nerdy people in there. Also, yo, someone needs Ross is in the Rockville area. If anyone's coming from there, you should give Ross a ride. Okay, he don't have a ride. He needs a ride. Again, Ross in there would be pretty clutch. So if you're coming from that area, he's in. Yo, Christmas miracle. Come on. Porcelio expansis. Oh, was that on there? I don't know. Well, the whole list of isopods she has are quite big. Porcelio expansis is cool though. Expansis. Yeah, they're very pretty. Very pretty. Thank you for another stream. Next, sleep already. Is the SP? No, none of these isopods are aquatic. Thanks for the deep dive. Appreciate you, JP. Trying to keep the work up. Uh, this is the aquatic isopod species, man. I need to get these guys settled in. Lurcius brachyurus, man. Come on. Come on. Anyways, yeah. So this Sunday, 1 p.m., North Haven Street, Baltimore, Maryland, 21224. Come through if you're able to. We got people coming from PA, coming from all over the place. Louise is driving 2+ hours to come and vend this thing just because she wants to hang out with all the people she's been talking to in the chat and stuff. Because honestly, yeah, a lot of people. We're just interacting on these streams all over the damn place, you know? So getting everyone in one spot in person is going to be pretty lit. That's LIT, in case you didn't know. Nick said I should start streaming every time you end yours just to keep and force you to stay awake. Jesus Christ. No, no, no. Look, I'm going to go to sleep. It's been fun. Hey, let me know in the comments, replay crew, or otherwise people who are here but need to contemplate. Let me know how you like these deep dive streams where we go through and attempt to read all this stuff that I don't understand. I feel like we still can get a good amount from it, and we get some fish to look up in the process. If there's any topics you're interested in me doing a deep dive or looking at, let me know. Otherwise, I'm gonna keep just finding whatever I find interesting, and if you know, good luck to you, whether or not you do. A couple of fish said, good night, everyone. Appreciate you. Thank you for stopping by, my friend. Mick said, take care, everyone. Happy December. Yes, take it easy. Take it easy on yourself. Enjoy as much as you can enjoy. Yeah. Mick said you're great at this, Mick. Nick, you're great at this, Nick. See, I just proved you wrong. I just proved you wrong. Always appreciate you, buddy. George said, "Okay, I'm back." Well, as usual, George, you're coming back as I'm getting off. All right. Zebra said, "Good night. Awesome stream, by the way." Okay, I appreciate it. Good. This is good. This is good feedback. George said, "Ready to party." George, George, the stream is ending, George. Oh my gosh, man. He has impeccable timing every time. I appreciate you stopping by. I'm gonna stop yapping. I'm gonna head off, and don't you dare forget. George said, "Okay, then I'll stream." Okay, check out George's stream then, guys. But yeah, don't you dare forget, man. Put me in coach. Never fly coach when I'm picking up the loach. Put me in the game. Swear to God, I'll rock the boat. Best sucker fishes, loaches get the vote. I'm gonna instigate. I'm gonna poke. Bears hate to see when we go live and push the yoke. I'm pushing the line. It'll be fine. We got the load. Pseudogastromyzon. We with the UFOs. All right. Holy. Thank you, Steve, for the $2 as we close out here. Said awful, very bad nerd stream. Okay, next time, Steve, make up more things other people said about me. I'll just go crazy and just like cancel myself. You know what I mean? Like I'll just say the worst things that could come to my mind possible. Like scud, 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 scuds. Okay, I'm headed out. Appreciate you guys. Good night. And this has been In My Tanks. Is that a hill stream loach? Or are you just a tasty snack waiting to happen? Night! And I'm